now mostly on bioinformatics, and she's also an expert in CAR T cells. And so we are working a lot now on systematic reviews and something that we can manage big data, and she's helping a lot. So probably, yeah, just wait 1 minute and then we can start.
Good morning, Dilton.
Rodrigo, I send you the PR number if you want.
Probably can start. I will try to contact him during the presentation of the lab, and let's see.
Rodrigo, we are already broadcasting, so
probably can start. I will try to contact him during the presentation of the lab, and Professor Vanderson, if you want to start, I think we can start, and I will try to contact Pierre during the presentation.
Okay, so I'm just going to put my presentation. Can you see it?
Yes.
Is it okay?
Yes, very nice.
So I would like, first of all, to thank all the speakers to join us in this Saturday. Here in Brazil is in the morning, raining morning in São Paulo, where I am in Belo Horizonte. I think everywhere is raining. And I'm very happy to have French colleagues here. And of course, this workshop was prepared by Rodrigo, Nalio, and Professor Barbuto. So I'm just going to present a general overview of our Laboratory of Medical Investigation that we call LIM here in the University of São Paulo. This laboratory is the number 31, and the main objective is to study the pathogenesis and target therapy in onco-immuno-hematology. So just to have an overview of the general hospital of clinics, is a complex of hospital. This is considered the largest in Latin America, a complex of hospitals. There are many areas that have been recently considered among the 200 areas in the world. So it's really important to tell you that we have many patients here, 2,400 hospitals beds, about 4,000 people, patients and employees that generally frequent our site. There are 6 medical specialties that are among the 200 best in the world. This is the classification of the world's best specialized hospitals in 2021. So the complex, we have many institutes, and we have the Institute of Cancer here called HC-FMUSP, that is a 24-floor building. And in total, there are 66 medical investigations laboratories associated with the Faculty of Medicine of the University of São Paulo. The main purposes, objectives of these labs are to develop scientific research, standardize new diagnostic techniques and methods, and to promote the formation of researchers and serve as a center for teaching and training students in health-related fields. Here is the organization of our lab. In fact, I am in Brazil since 2016, where when I took the leadership of the Department of Hematology. Previously, I was in Paris for 15 years in Hôpital Saint-Louis, and after I was 5 years in the University of Oxford. So my co-chair is Professor Barbuto, who is the vice director of this lab, and the main research field of Professor Barbuto is immunology of cancer and immunotherapy. So as you can see here, we have many direct collaborations with doctors and professors in our LIM. And just to tell you that we have Professor Eduardo Rego, who is a co-professor in the department, who in fact has a great experience in studies in the pathogenesis, diagnosis, and treatment of acute leukemias. And we have many others collaborators directly involved with this LIM, and in fact, more than 16 PIs, 23 collaborators and researchers, 2 postdoc, 20 PhD students, 14 master students, and 6 scientific initiation studies. Here is the main lab on cell therapy that I am directly involved. Rodrigo, who has recently joined us after PhD in Paris and a post-PhD in Paris, Theo, Samuel, Luciana, and recently more students, Felipe, Paulo, Fernanda, Viviane, Paula, and Natalia, who, in fact, I would like to thank Natalia for all the work she has done, because she is leaving us for her career. And the main lines of this research lab is the CAR T cells, CAR NK, CAR myeloid cells in cancer immunology, and gene therapy for sickle cell anemia. So, in fact, I would like to thank all my collaborators and all the team who is recently working hard with all the projects that we have. So, just I would like to congratulate Rodrigo and all the team. We have more than 240 participants in this workshop. You can see here, these are many students and medical doctors, biomedical doctors, biologists, and many other fields from many states of Brazil and also from outside Brazil. So I would like to wish you a very successful workshop, and I would like to ask Rodrigo and Barbuto to present our speakers and also to introduce the first talks that we are going to have during this 3 hours workshop. Thank you very much.
Thank you, Professor Vanderson. I prepared some introductory text to introduce our speakers. So I'd like to thank very much Pierre, now join us. Also, thank you very much, Pierre, Jérôme, and Christophe for your time. I would like to say that this is a gold opportunity for us in Brazil to have you in this meeting. And I think people from all the sides of Brazil, we really enjoy this. I will just introduce Dr. Jérôme Galon. So Dr. Jérôme Galon is Director of Research at INSERM and head of the Laboratorium Integrative Cancer Immunology associated to the Cordeliers Research Center and to Sorbonne Université in Paris, France. He directs interdisciplinary research programs on tumor immunology, mostly focused on the role of T cells and the importance of adaptive resisting immunity in human cancers. One of the main concepts developed by Dr. Galon and his group is the cancer immune contexture that provides the basis for the creation of the Immunoscore. So Dr. Galon has been recognized by several awards. It's a huge list, but I just need to highlight the William Collin Award. Also in 2019, he was the winner of the European Inventor Award. And in this year, 2021, he's within the top 1% of the most cited scientists cross fields worldwide. So I'd like to thank you very much, Jérôme, for your time. And it's an honor for us.
Thank you, Rodrigo. Thank you for inviting me. And I will share my screen. Do you see the screen?
Yes.
Yes. Good. I have to remove this thing. Oops. What happened? Okay. Do you still see the screen?
Yes.
Okay, good. So thank you, and I will talk about Immunoscore, tell you what it is in this new era of cancer immunotherapies. So here are my disclosures. I'm a co-founder of an Immuno-Oncology diagnostic company and chief scientific officer at LIM, that is now a Verasite company, and I talk to people in the industry. So just to set the scene, cancer is a very complex disease, and the tumor immune microenvironment is extremely complex because it's composed of possibly all the subpopulations of immune cells you can think of. And what we wanted to do is really to analyze and decipher and deeply characterize human cancers and get into the complexity of human cancers, not focusing on a particular molecule. I had no favorite cells to start with, but really to analyze this as a whole and do bioinformatics and systems biology in these human tumors. And by doing so, we put back in 2013 this review discussing and proposing this continuum of cancer immunosurveillance, depending upon the quantity and the quality and the immune response intensity, where the quality of the immune response and the intensity of the immune response could slow down tumor growth, protect again recurrence, and in the case of efficient therapies, get into immunotherapies in particular, get into tumor regressions. And we discussed the fact that there were predictive immune signatures associated with response to treatment, prognostic immune signatures associated with the good outcome of the patients, and mechanistic immune signatures, meaning what is increased after an efficient treatment. And these signatures, we discussed the fact that there is a common core of cells and pathways that are activated. And so we discussed this continuum of cancer immunosurveillance at all the stages, including on the left side before cancer, at the pre-cancerous stage. And I will briefly discuss this data. Continuum of cancer immunosurveillance, once a cancer is a carcinoma, is a cancer within the primary tumor. And then after dissemination, the continuum of cancer immunosurveillance up to the metastasis and even within the metastasis and at the metastatic stage. So why were we interested by looking at precancerous lesions? Well, these two findings that we published in 2011 and 2013 were pointing to the fact that the adaptive immune system and the densities of the cytotoxic T cells were at their highest densities in the smallest tumors, the T in situ tumor, T1 tumor, which are the first stage of carcinogenesis. This is when the patient had the highest densities of T cells, as well as many other adaptive immune cells, including memory T cells. And then it was decreasing over time and with the progression of the cancer, which meant that this adaptive immune reaction and the activation of the immune system with a memory phenotype could be set before the first stage of carcinoma. And so we wanted to analyze that and publish this paper, Immune Evasion Before Tumor Invasion in Early Squamous Carcinogenesis. So what we did is to take people at risk of developing cancer. So what is best than a heavy smoker to look at someone that is at risk of developing lung cancer? So we got access to bronchoscopy of heavy smokers. And on the tissue that were taken after the bronchoscopy, either we got normal lung on top, or we got already cancers at the bottom, squamous cell carcinoma, or we got tissue that were at all the possible stage between a normal lung and a cancer. So hyperplasia, metaplasia, mild dysplasia, moderate dysplasia, severe dysplasia, etc. And so we analyzed all these tissues to see what was coming and when the immune system was activated or was sensing that something was going wrong in the tissue. And just to make a long story short, I will not enter into this. This is a summary of the results that we got. So among those 9 different stages, from 0 normal to 8 cancer, in fact, from a molecular profiling with transcriptomic analysis of the tissue, clearly there were 4 major stages: the normal or close to normal, stage 0, 1, and 2, the low-grade dysplasia, stage 3, 4, and 5, the high-grade dysplasia, 6, 7, and then cancer. And what was happening within the epithelial cells was first some DNA repair deficiencies and increased proliferation, then metabolic changes in low-grade dysplasia, and ultimately, just before cancer, invasion and change with EMT-related genes. In terms of the immune microenvironment, we could detect an immune sensing with the resident immune cells in the tissue and an activation and unleashing of the response of those cells already in low-grade dysplasia. The most important changes in terms of immune microenvironment were happening at the high-grade stage before cancer. And in that case, simultaneously, there was a full activation of both the innate and adaptive immune system with a massive activation of the both T and B cells, acquisition of a memory phenotype, and then not only activation of the adaptive immune cells, but also multiple immune escape mechanisms, including increase of immunosuppressive cytokines, as well as immune checkpoint receptors, such as PD-1, PD-L1, TIM3, LAG3, etc., which were happening before the cancer stage. And so this opens new opportunities and led us to understand what was going wrong. So now, once a patient has a cancer, what is the clinical practice? What the clinicians are doing when a patient is coming at the hospital? Well, first they diagnose the cancer and they classify the patients. And all the classifications on the left that are done in routine clinical practice worldwide are in fact tumor cell centric, tumor cell related classification. So it's, of course, the tumor extension and invasion, the TNM staging system, tumor node metastasis, to progression into the primary tumor and invasion into the nodes of the distant metastasis. It's based on the tumor morphology, the anatomopathology, the tumor grade, the grade of differentiation, etc. On the tumor cell of origin, it's based on tumor molecular pathway that patients can be classified. Is it a microsatellite unstable tumor, for example, MSI, based on tumor gene signatures, or based, of course, on the tumor mutation status? Has the patient has a KRAS mutation or BRAF mutations, etc. But until very, very recently, because there was nothing in 2019, and nowadays we have Immunoscore, and I will discuss about it, but there was absolutely no immune evaluation of a cancer patient. When a patient is coming at the hospital, the clinicians do not know what is the immune response of a cancer patient, what is the host immune response, what is the immunity of these patients. And so we wanted to push to have a first immune-based classification of cancer patients. So really, what's the importance of these immunity, pre-existing immunity? This question, back in 2000, not solved, and there was a century of controversies about the importance of the immune system. Nowadays, of course, with immunotherapy, everybody agrees, but it has not always been the case. And this is just as a reminder, I believe the first publication putting a link and association and relation between lymphocytic infiltration within tumors and longevity of patients was from 1921, so exactly 1 century ago. And so it took basically 1 century to get Immunoscore, or an immune-based test, into routine clinical practice. And this Immunoscore came after our paper that we published in Science back in 2006, where we put this new concept, the cancer immune contexture. And we define the immune contextures are the important immune parameters that are associated with the survival of the patients. And they consist in 4 types of parameters, 4 different parameters. The types of immune cells, which immune cells are associated with the survival of the patients, the density of these cells, the location of these cells, the spatial information, and the quality of the immune response with immune signatures. And these 4 parameters are defining the immune contexture. From that, we derive the Immunoscore, which is a test that takes into account the first 3 parameters: the type, the density, and the location of immune cells within the tumors. And when we analyzed, in that case, the cytotoxic and memory T cells, patients with a high Immunoscore here have high densities of cytotoxic and memory T cells in 2 specific areas of the tumor: the center of the tumor and the invasive margin of the tumor. And as you can see with these thin curves on top, the patients have a very prolonged survival, more than 15 years follow-up here, and more than 80% of the patients not having a recurrence if they have a high Immunoscore. What is very important to see here is that this is happening regardless of the stage of the patients, and the patients having a more advanced stage would have the same good outcome if they have a high Immunoscore. This represents these, now that we call inflamed tumors or hot tumors, the strong pre-existing adaptive immunity, the patients having a high Immunoscore. In contrast, the thick curves are dropping very rapidly. These patients have low densities of cytotoxic memory T cells in the center and invasive margin of the tumors. And as you can see, these patients have a very bad outcome. These cold tumors, again, regardless of the stage of the patients. So even a patient at an early stage would have a very rapid tumor recurrence. And so this coordinated adaptive immune reaction, Immunoscore, more than the tumor invasion parameters, was predicting the clinical outcome of the patients. And really the novel paradigm came when we did the Caux multivariate statistical analysis to test the strength and the dependency of the parameters. And we put into the model the strongest parameters, so the T stage, the end stage, the grade of differentiation, together with Immunoscore. And in fact, as you can see, Immunoscore not only is highly significant on ECOX multivariate analysis, but beyond that, the other parameters are now no significant anymore. They are statistically dependent upon the Immunoscore of the patients, which means that the tumor progression, the tumor invasion is statistically dependent upon the pre-existing immunity of the patients. So the immune contexture looks at the cell, the types, the quantity measured by the densities of immune cells, look at the location, this is the spatial information, and look at the immune functional orientation of the response, especially a Th1 type for the T helper cells is very good, and this represents the quality of the immune response. We had multiple consecutive papers in different cohorts looking at these T-cell infiltration, and I had this quite provocative at the time editorial at the bottom here when we published one of our papers in GCO in 2011, TNM staging in colorectal cancer. T is for T-cell and M is for memory, which is what we are describing in our article. So we have different types of patients. We have the one on the right here, Immunoscore 4, full of cytotoxic T cells in the center and invasive margin. If we look at the median overall survival of these patients, they will survive more than 15 years, regardless of any other parameters. If we take patients with an Immunoscore 2, they will survive 5 years. If we take the patients with Immunoscore 0, these patients will survive less than 2 years. So we are talking about major clinical differences. And so in 2012, we decided to initiate a worldwide effort to try to have a consensus Immunoscore and to validate it globally. And this worldwide consensus Immunoscore study had a lot of centers and countries who wanted to participate. And after a lot of assays, harmonization, so the study was designed in colon cancer patients with stage 1, 2, and 3. And the study design was the following. All the center after the harmonization, so we spent a long time first to decide what should be the Immunoscore, what should be the markers, how we should quantify them. And then there was assay harmonization so that each center would perform the Immunoscore on their patients. The raw data were sent for QA/QC and then were sent to an external statistician at the Mayo Clinic outside of the consortium. And then in parallel, all the centers sent their clinical data to a US site who encrypted the data and sent them to the external statistician who divided into 3 independent cohorts and validated the Immunoscore, which is a method with standardized operating procedures using automated machines, high-resolution scanners, and digital pathology with an Immunoscore software that counts every cell on the tissue to give the full densities of the cells and to transform them into the Immunoscore classification. So it's a full slide quantification. All the cells are counted. It's not subjective field. It's with predefined cutoff, and it's a quantitative immunopathology assay. So the results of the study were published in The Lancet in 2018, international validation of the consensus Immunoscore for the classification of colon cancer. These are some raw data where you see here the densities of the T cells in the center of the tumor, and we have the same at the invasive margin and for the CD8 cells. And you can see that there is a huge density of T cells inside the tumor. Each dot is one tumor from one patient here, more than 3,000 patients. And you can also see the huge difference between patients. There's a lot of variability between the patients with more than 3 log 10 difference in the densities found inside the tumors. And when we look at the clinical results, Of course, the primary objective and all the secondary objectives were reached. Immunoscore stratified the patients into risk categories. This test is the relative contribution to the risk. On the left side, you have the current staging system, where you see the contribution of the different parameters. N stage is the most important, followed by T stage, rate of differentiation, VLP, venous emboli, lymphatic invasion, perennial invasion, macrosatellite instability. When we add on the right side Immunoscore, you can see that Immunoscore represents half of the risk, higher than all other parameters. But on top of that, and even more importantly, on the multivariate analysis, all the other parameters are in fact statistically dependent upon the Immunoscore of the patients, so that on multivariate analysis, whether we classify patients into 2 Immunoscore groups, high, low, 3 groups, high, intermediate, low, or 5 groups, Immunoscore 0, 1, 2, 3, 4, all p-values on the multivariate are below 0.0001. So this is for all stages of the patients. What about at each stage? Because of course, clinicians, when they see a patient, they have one patient at a given stage. So clinicians are interested to know, is the Immunoscore, is a biomarker important at a given stage? So this is the analysis for the stage 2 patients. And just as a reminder, we already published back in 2009 this GCO article focusing on the early stage colon cancer, which are the stage 2 colon cancer. And we also looked at, and so we validated this aspect on all stage 2 patients. Clinician wanted more and asked for the high-risk stage 2 patients, which are the T4 and 0 patients. And so we focused an analysis on the T4 and 0 patients. And as you can see, these are supposed to be high-risk stage 2 patients. When they are stratified based on Immunoscore, with the predefined consensus Immunoscore, on top you have the patients with a high Immunoscore, meaning high densities of cytotoxic T cells. And as you can see, these patients, in fact, have a good outcome. The only high-risk patients are the ones who have a low Immunoscore. And so this shows that the definition of the risk, even within an existing subgroup of patients, is very much impacted by the Immunoscore of a patient. And similar results were found if we focus the analysis on untreated patients. The multivariate analysis showed that putting all the parameters together, only Immunoscore remains significant on a Cox multivariate analysis. All the other clinical parameters became non-significant with an hazard ratio 0.15, and the relative contribution to the risk compared to all other clinical parameters showed that, in fact, Immunoscore represented 76% of the risk explanation. What about stage 3? Stage 3, so these patients are treated with chemotherapy. This is the recommendation for these locally advanced colon cancer patients. So now we have 4 independent studies and cohorts representing more than 2,000. And this is a very large international effort that was finally published into the New England Journal of Medicine, even though the primary objective of that study was not reached, and the study was, from the primary objective standpoint, unconclusive. So the study was to see whether 3 months of chemotherapy was non-inferior to 6 months of chemotherapy. So as you can see from the curves, they are not statistically different, but they are not statistically similar as well, which means that it was impossible to from a statistical standpoint, to say whether 3 months was non-inferior to 6 months, even though the curves were very close. If we look at this cohort of patients with the IDEA France study on more than 1,000 patients, these are the patients, when you stratify them based on their Immunoscore, now you can see major differences between the patients in terms of their outcome. And Immunoscore can predict really high-risk patients and low-risk patients even on that population. But what about the randomized trial, the 3 months versus 6 months? Does the pre-existing immunity has an impact on the likelihood of response to chemotherapy? And the answer is yes. Here you have on the left side the patients having a high Immunoscore. So all these patients were treated either on the randomized 2 arms with 6 months in white or 3 months in blue of chemotherapy. And as you can see now, if the patients have a high Immunoscore, they do significantly benefit in receiving 6 months chemotherapy with a very good hazard ratio and significant p-value. In contrast, the patients with a low Immunoscore did not significantly benefit in receiving 6 months chemotherapy. These results were validated as well in subgroup analysis, including within the low-risk stage 3 or within the high-risk stage 3 patients. So Immunoscore in colon carcinoma, we also published real-life patients, so patients that are just treated with standard of care or in real life, not within a randomized phase 3 clinical trial. And in this GCO article in 2020, we showed that Immunoscore was not impacted by aging, that you can have a very good Immunoscore and be full of T cells even if you're 90 years old. Immunoscore is, of course, a prognostic marker and it has the highest contribution to the risk. And we also validated that Immunoscore predicted the response to chemotherapy in that setting. So same types of curves, Immunoscore, high Immunoscore on the left. Now we are comparing patients that received chemotherapy in blue or did not receive chemotherapy in white, just because they refused chemotherapy or they didn't do chemotherapy for whatever reasons. It's not a randomized, these are real life patients. And you can see that there is a significant benefit in having chemotherapy when they have a high Immunoscore. But the patients with a low Immunoscore here, as you can see, do not significantly benefit in receiving chemotherapy. So we were talking about locally advanced cancer. What about the metastatic stage? Is there an immune escape at some point? So for that, we analyzed several cohorts, including these 2 studies here. So we took primary tumor colorectal cancers and analyze the metastasis, the resected metastasis from these patients, either liver metastasis or lung metastasis. And we analyzed more than 600 metastases that were fully resected in order to deeply analyze the immune microenvironment of each of these metastases. I will not enter into the details of this publication. I will focus on another paper and another study that we did on the metastasis to see, what are the metastatic immune escape mechanisms and what is it compatible with the current theories of cancer evolution. So just as a reminder, the theories of cancer evolution, how a cancer is disseminating, there are 4 models that have been proposed. The linear model, saying that you need the accumulation of different driver mutations in order to get dissemination and the evolution of cancer. In contrast, the neutral model says that there's no selection whatsoever of any particular mutations, and this is a neutral evolution. In contrast, the Big Bang model says that you need to have multiple driver mutations before dissemination can occur. Probably the most accepted model is the branched model, saying that you can have on different tumor clones, different driver mutations, and that these will evolve in parallel. What is common between these 4 models is that none of them involve a possible role of the immune system and a possible Darwinian selection of tumor clones with the immune system. So these theories are cell-centric, and none of them involve the immune system. So we wanted to address this in human, and we published this article in Cell: Evolution of Metastasis in Space and Time is Under Immune Selection. So what we did in order to be able to analyze this was to take patients having a primary tumor, having multiple synchronous metastases, in order to be able to analyze these synchronous metastases, and having multiple metachronous metastases, and all these metastases being resectable, that the clinician, the surgeon, was able to resect and to remove so that we could fully analyze them. And so we selected patients based on these criteria and got patients who had more than 11 years of cancer and follow-up with multiple recurrence and multiple metastases, and did multi-omics technologies, genomics, RNA sequencing, exome sequencing, multiple assays, and multiple immunomics technology with the immune microenvironment analyzed with multiplex assays. And this is an example of, after having done the phylogenetic tree, we did the evolvogram of the tumor cells, the tumor clones. So what you can see here first are on the left side, the P is the primary tumor. We can detect based on the sequencing, the exome sequencing, the clones. So the major clone in that case is the blue clone. But you can see that this blue clone is disappearing and is never transmitted to any of the metastases thereafter. The clone that is transmitted is this purple clone that is not the major clone on the primary tumor. This purple clone is transmitted, but it's not the major clone anymore already on the first synchronous metastasis. Is the orange one, which has a subclone inside it, the green subclone. And you can see that this green subclone is not transmitted, but the orange one is transmitted. And by doing so, we could look at the evolution and the evolvogram of the tumor clone. And we analyzed deeply the types of mutations they have, each of these clones, and found that, in fact, the non-recurrent clones are immuno-edited, whereas the progressing clones, those that are transmitted to a next metastasis, are immune privileged. So we looked at the immunomics, we looked at multiple immune parameters within each of the metastases. Here I'm showing you two parameters, the Immunoscore of the metastasis. So within each metastasis, we perform the Immunoscore, the densities of the cytotoxic T cells inside the metastasis at the invasive margin of the metastasis. And we are plotting the immunoediting score. So the immunoediting being the likelihood of having a deletion of an immunogenic mutation compared to a silent mutation that is not seen by the immune system. And you can see the different metastases here that are plotted. And you can see that for immunoediting to occur on those patients, a high immunoscore is necessary. So the more immunoediting there is, the lower is the value. So this increased immunoediting goes with a decrease in the value. And the immunoscore is not sufficient because you can see that patients with a high immunoscore can still get an absence of immunoediting. So it's necessary but not sufficient. And basically, I will skip many of the analysis to go into the conclusion. These three major types of mutations have different characteristics, and in fact are associated with particular defects. The low immunoscore, no immunoediting are associated with a loss of heterozygosity in HLA, in very important genes such as IL-15 and haploidy alterations. The high yes, which are in fact the ones where the best, the ones where there is no dissemination of those tumor clones. These tumors have the highest immunoscore. They have a strong proliferating T cells in their tumors, whereas the high no have a lot of immunosuppressive mechanisms associated with these metastases, including increased Treg cells, increased PD-L1 expression, etc. And we could show that there is a multiverse of metastases, that the evolution of the tumor clones is in fact linked with the local intra-metastatic immune contexture, and that the non-recurrent clones are immuno-edited, whereas the progressing clones are immune privileged. And in fact, we proposed an alternative model that is a parallel immune selection model, that in fact shows a dynamic interaction of tumor cells with the immune cells and a Darwinian selection of the immune escape variants with this parallel evolution and within this multiverse of metastasis. And we validated this in an independent cohort of more than 130 patients for the dissemination from the primary tumor to the first metastasis, these parameters. So Immunoscore now, after multiple studies and has now reached the point where the immune response has finally been included for the first time in the latest edition of the WHO book, the WHO classification of digestive tumors, as an essential and desirable diagnostic criteria for colorectal cancer, and stating the Immunoscore and the Lancet publication. And finally, in 2020 and in 2021, the ESMO European Clinical Practice Guidelines for Gastrointestinal Cancer and the Asian Clinical Practice Guidelines introduced Immunoscore into the guidelines, both for their prognostic value and the predictive value of response to treatment. So if you're more interested by learning more about the immune contexture, the Immunoscore in cancer prognosis and therapeutic efficacy, you can read this Nature Reviews Cancer that we wrote. And if you're more interested by immunotherapies, and I encourage you to read this other review in Nature Review Drug Discovery, where we proposed alternatives to treatment by categorizing patients first based on their immune status, and approaches to treat immune hot, immune altered, or immune cold tumors differently with combinations of immunotherapies, knowing the alterations and the pathway, and propose different alternatives. And finally, I have postdoc and also a project and lab manager positions available if some people would like to join my lab. And I would like to thank all the people in the lab who contributed, to thank all our collaborators, the clinicians, our sponsors, and also to thank deeply all the centers in the world who participated into the validation of the consensus Immunoscore. And I thank you for your attention.
Thank you very much.
Thank you very much. It's very inspiring presentation for everybody. I think all the scientists, independent of the field. I don't know if you want to start, Professor Barbuto, Professor Vanderson.
Yeah, I just have one comment and one question. Every time I look at your, I see you change the way oncologists look at immunology of tumors, because immunologists always consider the importance of the immune response, but the oncologists never believed. You show them definitively that has to be considered. And my question is, and what of the role of CD4 T cells within the tumors? Did you see anything?
They are very important. So when we are looking at the quality of the immune response, which is the fourth parameter of the immune contexture, the quality of the immune response, definitely CD4 T cells have a major role. So associated with the good immune contexture, you have the Th1 cells, so T helper 1. It is very important to have a tumor that is producing from gamma, and there are other subtypes of T helper cells that are important, and other subtypes of CD4 cells, in particular the T follicular helper cells, which are in particular in these tertiary lymphoid structures, and that migrates to the margin of the tissue. And these tertiary, these T follicular helper cells are helping also to set up this good immune contexture by producing specific cytokines and chemokines. We can cite two of them. CXCL13 is a very important one, and for the cytokines, IL-21 is a very important cytokine that shapes the good T cell response, both for the CD4 T helper 1 and for the cytotoxic memory T cells. So,
thank you. Martin has raised his hand.
Yes, thank you, Barbuto. Jérôme, fantastic talk, and I fully agree with Barbuto's assumption that the oncologists really never attached to this idea as a first moment, right? So it's very important to bring all this avalanche of results you're showing here. I'd like to ask you for a comment regarding some of your works, actually, and others, linking the immune contexture to the molecular lesions you can find in tumor cells, right? Because those leukocytes are there for some reason, the way they are there, and this is definitely shaped by features of the tumor and also features of the host, right? So I'd like you to comment, such as your results on IL-15 and many others with interferon. So will there be a point where we can just look at the molecular lesions in the tumor and the genetic characteristics of the host, including that maybe there may be the microbiota or so, and use this as a proxy for the Immunoscore or something like that?
So in fact, when you have a high Immunoscore that is associated with a good immune response and a good immune contexture within the tumor microenvironment, this is the results of many parameters, including, of course, the tumor cells and the tumor cell aberrations, let's say. It can be mutations, but not only mutations. So the work I showed you on the Cell paper with this metastatic dissemination and the relationship between the immunoediting of the tumor clones clearly shows that if we concentrate on the mutation, on the types of mutations, so not only the mutational load, but the types of mutations, whether they are immunogenic or not immunogenic, matters to set up a good immune contexture or a less good immune contexture. So we know that there is this relationship, and indeed we and others as well have shown that you have T cells that are specific against specific mutations, in particular the frame shift mutations, those that are changing the amino acid because of a frame shift in the DNA. These generate very immunogenic peptides, and we can detect T cells against these frame shift mutations and show that you have very functional T cells that can kill tumor cells if they have that particular mutation. But the mutations is not everything. There are many other changes in the tumor cells, many stress signals, many overexpression of different genes that are aberrantly expressed on tumor cells that can be presented and recognized as well by the immune system. So it's, in fact, the immune contexture and the cytotoxic T cells is the endpoint of all these possible alterations that we can measure in very complex ways by other means. But basically, if you look at mutations, you will not consider other alterations. And so I believe that it's in fact way more powerful to look at the ultimate fighters, which are the cytotoxic T cells, rather than to try to search for proxy, because there are multiple proxies that you need to put into the equation, basically. We also have published this year with Davide Bedegnetti a paper looking at the host, so the patient, the polymorphism. We are not all equal. And so we found that there are indeed some genetic polymorphisms that are associated with a better likelihood of having a good immune contexture. But I think this is representing a minor point in comparison to all the aspects of the tumor biology.
Sure. Much easier to go straight to the final result. just because it's intriguing in a mechanistic way, the different layers that will impact the final outcome. So it's very nice to see work being done and showing these different layers. So thank you again, Jérôme. Thank you.
Thank you.
Professor Vanderson, yeah.
Wonderful talk, Jérôme. Merci beaucoup. I was really impressed. You know, we do not have experience on solid tumors, cancer, because it's more hematology. So my question is first, which other cancers you have detected the Immunoscore importance? And what about hematologic cancers such as lymphomas, leukemias? What do you think about the Immunoscore? And the other question is, if the Immunoscore is so important, why you think that CAR T cells has not been working in solid tumors?
Yes, so I should have put a few slides on the hematological malignancy, because we have actually we got data on DLBCL, so on diffuse large B-cell lymphoma, where we analyze the immune microenvironment and look at the impact of Immunoscore, and also look at the quality of the immune response with this immune signature, so immunosign, and in the context of CAR T cells, actually. And in fact, what we found was that it's not only about the CAR T cells themselves, about the CAR T cell constructs and the types of CAR T cells you infuse to the patients. In fact, the CAR T cells are working or not working depending upon the immunity of the patients. And we showed that the quality of the immune response, the immunosign, and the immunoscore that we can measure in the tissue is very much associated with the likelihood of CAR T cells to expand and to be efficient and to protect the patients and to prolong survival or non-prolonged survival. So it is the case that the pre-existing immunity in liquid tumors, at least in DLBCL, in lymphoma, is very, very important, not only for the disease, but also for CAR T cell treatment. So we analyzed, for the hematologic malignancies, these are the main results that we have. This has been shown in several meetings and hopefully will be published soon.
Thank you.
I think Professor Dilton also has a question.
Thank you very much. Nice talk, Jérôme. Fantastic talk. I think, in my point of view, the immune system exerts its pressure over all threats that are presented to it. And I think that external enemies, such as infection by viruses, fungi, bacteria, and so on, as well as internal pressures like exerted by neoplastic diseases, make an imbalance of the relation, the microecological relation on the immune system and the enemies. And I think that the cancers are the most clear enemy that drives an ecological disturbance on the system of the immune system, and also as on the tumor cells that must evolve in order to get more fit to escape the immune system. I think it's very, very interesting your idea.
Yeah, I agree, and also the immunity and the relationship between these neoplastic cells and tumor cells and all these alterations that the immune system is detecting is different in each of the metastases. We were able to analyze multiple metastases and multiple synchronous metastases in the same patients, and they all have a different set of tumor clones with different mutations, different tumor gene expressed. So it's very heterogeneous on the tumor cell side, but it's also very heterogeneous on the immune cell side, which means that there are different immune response in each of the metastases, which makes it so it's very dynamic, and it makes it very complicated. Once the patients reach the metastatic stage, the immune system has to deal with multiple fights at the same time.
Very nice.
Thank you very much.
We have time for the last question from people from YouTube. I will just ask Samuel to.
Hi, yeah, we have a question from Maria Audicia Moraes. She's asking if there is any known relation between the Immunoscore and molecular biology profile, like in colon cancer and patients with CRAS and NRAS gene mutations.
So yes, there has been some publications and correlations that have been made between molecular features of the tumor cells and the Immunoscore. So can cite probably the strongest, one of the strongest clear correlation is with the microsatellite instability, or the mismatch repair deficiency patients. They have, because of this mismatch repair deficiency, accumulation of frame shift mutations, which are very immunogenic. And so very often, not always, but very often these patients have increased T cell because they recognize these frame shift peptides. And so frequently they have a high Immunoscore. When these patients do not have a high Immunoscore because they have other immunosuppressive mechanisms in place, if these microsatellite instability patients have a low Immunoscore, they will not do well and they will have a poor outcome. But in contrast, the majority of the patients are stable. They don't have this mismatch repair deficiency. And even though some have a high Immunoscore and some have a low Immunoscore. So there are many other tumor alterations, tumor mutations that have an impact on the immune system and on the Immunoscore. But as I said earlier, this is very complex. There are multiple tumor molecular features of the tumors that are impacting either positively or negatively on the T cells. And so there's been some publications, notably on the TGF-beta pathway that is associated with a lower Immunoscore. There are some publications showing that the Wnt beta-catenin pathway is more associated with desert-type cold tumors. So there are different pathways that have been linked with differences in cytotoxic T cells and in different Immunoscore. But many of them, many alterations.
Thank you very much. I think we need to move forward. So thank you very much again, Jérôme, for your presentation. It was fantastic for us, very inspiring. And we hope we hope to see you soon here in Brazil when things are better.
Yeah. Hopefully COVID will permit.
Okay.
Thank you.
Thank you. So I will introduce our next speaker. It's also an honor for me to introduce Dr. Christophe Caux. Dr. Christophe Caux is Director of Research at INSERM within the Lyon Cancer Research Center in France, where he directs the Cancer Immune Surveillance and Therapeutic Targeting team. The team conducts fundamental and translational research projects aiming to identify immune escape mechanisms and develop therapeutic strategies to restore anti-tumor immune responses. Dr. Caux has key contributions in the human dendritic cells field and has pioneering studies describing the mechanism of immune escape in breast cancer. Dr. Caux published more than 200 scientific articles and participated in more than 25 patents. It's my honor to introduce you. Thank you very much for your time.
Thank you, Rodrigo. Okay, can you see my screen?
Yes.
Yes.
But
you can probably change the
projection. We're seeing.
Yeah, fantastic. Yes.
Okay.
Nice.
First, I would like to thank you, Rodrigo, and Dr. Barbuto for the invitation and proposing me to be with you today. So I am directing a team named Cancer Immune Surveillance and Therapeutic Targeting within the Cancer Research Center of Lyon, directed by Patrick Mellen. And today I'm going to, from what I understood from Rodrigo, to explain my contribution within the field of dendritic cells from physiology to escape in breast tumor. I'm going to discuss contribution in the adaptive immune response, the role of dendritic cell in adaptive immune response, innate immune response, some contribution in the heterogeneity of dendritic cell subset, and I will focus on more recent data of the impact of the tumor on dendritic cell function. And I will end up by some conclusion regarding manipulation of DC for immunotherapy. So this is the life cycle of dendritic cells that we understand today. So dendritic cells are from bone marrow origin. They originate from CD34 progenitors from the bone marrow. They circulate as immature dendritic cells through the blood, and they colonize every tissue, most tissue, where they form a network of rare cells, where they play a role of sentinel of the immune system. They will, in this tissue, sense the danger and capture antigen, and when they are activated, when they sense the danger, they will emigrate through the lymphatic stream into the draining lymph node, and in the draining lymph node, they will encounter naive T cells that enter through the bloodstream, and they will present the uptake antigen to the naive T cell and induce priming of the T cell activation. This is a very important step to initiate immune responses. And my contribution to the field starts from the observation that GM-CSF cooperate with TNF-alpha to induce dendritic cell development from CD34 progenitors. And this was in 92, and this was the first step, the first model allowing to generate quite large number of dendritic cells and open windows to opportunities to study dendritic cell biology. And we use this model to characterize function of these cells. In particular, we were interested in the migration capacity of these cells, and we show that immature dendritic cell can colonize tissue, in particular epithelial tissue, through sequential recruitment through chemokine gradient, in particular through CCR2 and CCR6 ligand. CCR6 ligand meets through alpha CCL20 now. So we also show using this model that dendritic cell exists in two different stages of differentiation. They are immature, called immature in the periphery, in the tissue, in the non-lymphoid tissue, while they are mature, activated cells in the lymphoid tissue. And the signals that trigger their activation and their maturation are danger signals such as TLR ligand, for example. And they move from an immature state where they are specialized in antigen uptake into a specialized stage of of T cell co-stimulation through upregulation of class 2 molecule and co-stimulatory molecule. And this activation step triggers emigration receptors involving their maintenance in the peripheral tissue and acquisition of CCR7 receptor that is induced upon activation of the dendritic cell. And this is very important for the emigration through the lymph and for the enter into the draining lymph node through both CCL21 and CCL19 chemokine. And it is very important to understand that the T cells that enter in the lymph node also through the blood also use CCR7 to to meet together with dendritic cells. So the same chemokine and chemokine receptor allows interaction between DC and T cells to induce the first step of T cell activation. So then when the T cells meet the dendritic cells, there is several signals that are transmitted by the dendritic cell, and there is a crosstalk, a bidirectional crosstalk between the dendritic cell and the T cell. The first signal is the engagement of the TCR through the MHC class 1 or class 2 complex. This signal will engage ligand receptor interaction that we will call to the immune checkpoint. The first step is the upregulation of CD40 ligand on the T cells that will engage CD40 on the dendritic cells. And this CD40 is a very important signal to induce DC activation, induce several processes. The first one is the upregulation of co-stimulatory molecule like CD86. And we contribute to show that CD86 is a ligand for CD28, and those engage CD28 induce signal 2 on T cell and thus induce full activation of T cells. CD40 not only induce this signal 2, but also induce signals 3, such as IL-12, which is very important in dendritic cell to drive Th1 differentiation leading to interferon gamma production. So not only dendritic cells are critical to induce this first step of T cell activation, but we also show that they can contribute to activate B cells through this crosstalk with T cells. And in particular, they induce B cell proliferation and B cell differentiation into plasma cells, producing immunoglobulin, and IL-12 was shown to be a critical cytokine to this within this process. The dendritic cells are key cells to induce priming both T cell and B cell immune responses. So this was our contribution regarding the role of DC in physiology in the immune response, and I will summarize some observations regarding the role of DC in innate immune response interacting with other cells, in particular NK cells. And I'm not going to detail this, but just the concept that we proposed at that time, that for a T cell or an NK cell to be fully activated, you need the immune system needs to be alerted through at least two pathways at the same time. And one example is the activation of PDC through TLR7 and the activation of the CDC or MDC through another TLR, such as TLR8 or TLR3. The two signals are needed for cooperation, leading to type 1 interferon that is needed for IL-12 production to activate NK cells, for example, or could be two signals on the same DC or two signals on two dendritic cell population or different population, or a signal on the DC itself and on the NK or on the T cells. So this is a quite important concept that you need for the immune system to be alerted, to have different ways to alert the immune system to have a full effective response. I'm blocked here. Okay. We also have some contribution on dendritic cell subset heterogeneity. We now know that dendritic cell population is quite heterogeneous. Uh-huh. I don't know what happened. I'm blocked. I'm sorry, I can.
If you use the keyboard, probably.
That's what I'm doing. I'm frozen. I cannot escape.
Cannot arrêter le partage, no? And you can restart. As the computer probably.
Just leave the full screen. Yes, it's okay.
Yeah, it's okay now.
I hope it will be okay for the next slide. I'm sorry for that. Okay, so we know that there are at least two major populations: the conventional dendritic cells, or the myeloid dendritic cell, and the plasmacytoid dendritic cells. So the myeloid one, or conventional, are mainly involved in bacterial recognition, or at least they were believed to be involved mainly in bacterial recognition through cell surface receptor, like toll-like receptor 2 and 4, but also the question of toll-like receptors 3 and 8 within the endosomal compartment. They are mainly involved in IL-12 production and have high antigen uptake capacity and presentation capacity. In contrast, the PDC do not produce IL-12. They produce mainly type 1 interferon in response to virus recognition, nucleic acid recognition from viruses through TLR7 and TLR9. So they are critical cells in antiviral immune responses, and they have the lowest antigen uptake and presentation capacity. Sorry, this is still... So you don't see my screen?
Now we see, yes.
Do you see in the presentation mode or no?
No, that is in PPT model, yeah. Yes.
Is it okay?
Yes, great.
I hope it will not be problematic again. And within these two populations, so PDC is a quite homogeneous population of cells, while the myeloid ones is an heterogeneous family of cells, and we contribute to show that in these CD34-derived cells, GM-CSF is leading to the differentiation, to the development of at least two lineage of cells: the Langerhans cells, which are the epithelial cells of the dermis, and what we call interstitial dendritic cell, dermal dendritic cells from the dermis. And regarding PDC, we were interested to analyze their migration route. And while myeloid cells or conventional cells will colonize a peripheral tissue, PDC are mainly found in blood and in lymphoid tissue, and we contribute to show that they are recruited through sequential chemokine gradient in cooperation between homeostatic chemokine like SDF1, CRCL12, and ligand for CRCL3 like IP10 or MIG. And this cooperation was also involved in the recruitment during inflammation. They are not present PDC in homeostasis in peripheral tissue, but they can be recruited into inflamed tissue. And this chemokine cooperate with also other inflammatory chemokine like CCL20, CCR6 ligand for recruitment into epithelial tissue. So clearly PDC and MDC have different route of migration, but they can still, upon inflammation, reach environment. Okay, that was quite historical work on the physiology of dendritic cell subset, and more recent work have been made in the team regarding the role of dendritic cell and the role of the tumor environment impacting dendritic cell function. So this is the same scheme presented in another way, illustrating the different stage of dendritic cell dendritic cell life cycle, the development, the differentiation, the antigen uptake within the tumor environment, and the emigration into the draining lymph node. Each of these steps can be altered in the tumor environment, and in particular, we initially show that in the tumor environment, the tumor can shift dendritic cell differentiation or alter their function. This was made both in human and in mice models. And what I would like to focus in on two more recent observations regarding PDC and CDC1 function. So I'm going to summarize the observation we made on PDC's plasmacytoid dendritic cell. So the principal function of PDC is to produce type 1 interferon. And we show that in the tumor, in particular in breast tumor, that the function of PDC, this function to produce type 1 interferon, is impaired. They cannot produce type 1 interferon anymore. And this is in particular mediated through TGF-beta produced tumor environment. This empowers PDC to amplify regulatory T cells and to induce IL-10 producing T cells. And we show that this amplification by PDC was mediated through ICOS, a co-stimulatory molecule expressed on Treg in the tumor environment, and PDC expressed ICOS ligand. And we propose ICOS as a therapeutic target to neutralize Treg expansion in breast tumors. So this regulatory T cell and IL-10 producing T cell expansion represent two arms of the immune system that will block the effector cells, CD4 and CD8 T cell, allowing tumor progression. So we believe that in breast tumor, PDC play a central role in an immune escape network driving Treg expansion. And if we can find ways to restore type 1 interferon by PDC, this will allow to prevent regulatory T cell expansion and IL-10 production. So finding ways to reactivate PDC in the tumor environment can be also a strategy to restore tumor immune response. Now I would like to discuss some recent observation on CDC1. So CDC1 is another subset of dendritic cells that is found in blood, that is also bone marrow derived. This is probably the smallest population, less than 10% of the dendritic cell in the blood or in tissue. They have been shown, they have been well characterized in mice, and shown in mice to be critical cells for presentation of antigen to CD8 T cells. So just a few words regarding cross-presentation. So classically, there is two classical pathways of antigen presentation. The endogenous antigen in any cell will be presented in class 1, while on antigen-presenting cells, exogenous antigen will be presented into the CMH2 pathway. A unique feature of dendritic cell, at least some population of dendritic cell, is to mediate cross-presentation, presentation of exogenous antigen to the class 1 pathway. And this is a critical pathway to induce a primary immune response against virus-infected cells or tumor cells. So this is a very important process for anti-tumor immune response, to present antigen to CD8 T cells. And CDC1 have been shown both in human and mice to have a unique, an exacerbated property to mediate cross-presentation. They are not the only one, but they have a specialized function that's probably linked to specific receptors that they express, such as CLEC9A, which is an endocytic receptor allowing to endocyte apoptotic cells. They are also equipped with a series of receptors allowing the interaction with effector cells, T cell and NK cells. And in particular, they express the chemokine receptor CXCR1, which allow interaction with T cell or NK cells that express the ligand. And they also have unique features such as the expression of TLR3. They express this double-strand TLR receptor, double-strand RNA TLR receptor, and they also have the capacity to produce type 3 interferon lambda, while PDC produce type 1, CDC1 produce type 3. And we initially show that CDC1 have a unique capacity or have the capacity to interact with NK cells and through the production of IL-12, they will activate NK cells that in turn will produce huge level of interferon gamma, and this induce the cross-presentation, activate the cross-presentation capacity by CDC1, allowing to present antigen from tumor cells to CD8 T cells. So we believe there are important signal received during the innate immune cell activation to induce priming of the CD8 T cell response. And so this leads us to investigate more on this population of dendritic cells. In collaboration with Marc Dallot, that is working in mice, they developed a mice deficient in CDC1 using the XR1 promoter coupled to diphtheria toxin production. So these mice are devoid of CDC1. And we use a mammary tumor model, a triple negative mammary model implanted in mice that is very immunogenic and do not grow in wild-type mice. But in contrast, this tumor developed in CDC1 deficient mice. So clearly showing that CDC1 are involved, are critical cells in tumor immune surveillance. If they are not there, the tumor can develop. So this leads us to investigate the CDC1 in human tumors. And this is by flow cytometry within breast tumors. We see that there is a selective increase in CDC1 in the tumor environment compared to the other DC and compared to the blood. And these cells colocalize with tumor area enriched in CD8 T cells. By using transcriptomic database and specific signature for CDC1, we show that CDC1 are a good prognostic impact in breast tumor here. And this impact is also observed in multiple other tumors. And this was confirmed at the protein level using tissue section by multi-immunofluorescence here in a cohort of triple negative breast tumors, where we also confirmed that CDC1, high density of CDC1 is a good prognosis factor in triple negative breast tumor. And we have been interested in looking at type 3 interferon because it's a unique property of this CDC1. And we observed that within breast tumor, there is about a third of the tumor where we can detect spontaneously constitutive endogenous production of type 3 interferon by CDC1. And this is only produced by CDC1. The other DC do not produce, the other immune cells or non-immune cells do not produce type 3 interferon in breast tumor. And this was confirmed by multi-immunofluorescence, by double hybridization for interferon lambda in green and CDC1 marker CLEC9A in red. So in situ, only CDC1 produced type 3 interferon and not in all tumors. We analyzed, still in the transcriptomic database, the impact of the expression of interferon lambda or its receptor in outcome in breast tumor, and we see positive impact of both the ligand and the receptor in breast tumor. So this leads us to propose that CDC1 are important in tumor immunosurveillance through their capacity to produce type 3 interferon. So this leads us to investigate what is the role of type 3 interferon, what is the target of type 3 interferon. And here I'm just a preliminary data, and we have focused on immune cells, and this is total PBMC, looking at the impact of type 1 or type 3 interferon on immune cells by looking at phosphorylated STAT1, which is done with the team of the receptor of type 1 and type 3 interferon. So there is many cells that respond to type 1 interferon in the blood. Most cells respond to type 1, B cells, T cells, monocytes, dendritic cells. In contrast, in response to type 3, we see a tiny population of cells responding to type 3 interferon. And these are all plasmacytoid dendritic cells. So this clearly suggests that there is a crosstalk between CDC1 and PDC in the tumor environment. And we further analyze in situ the distribution of the cells. So this is multi-IF on tumor section in breast. So we here see that not only CDC1 interacts with CD8 T cells, so CDC1 are in yellow and CD8 in red here. So we see with the star there is many sites of interaction, but they also interact with PDC. So the PDC here are in red and CDC1 in green, and the star again illustrates the interaction. So we see in some tumors, many interaction sites between CDC1 and PDC. So this suggests that there is a crosstalk between CDC1 and PDC, and what is the consequence of this interaction in terms of immune surveillance and patient outcome is something that we are currently investigating. But we are really looking at the role of type 1 and type 3 interferon and the cooperation between PDC and CDC1, as well as interaction with other innate cells in tumor immune surveillance. And we are focusing in breast tumors, comparing early stage of tumor development, normal tissue, hyperplastic tissue, in situ carcinoma, and invasive carcinoma. And this is done through single-cell RNA sequencing and combined to spatial analysis in the objective to define a network of interaction through tumor progression in the objective to identify immune surveillance mechanisms involving dendritic cell populations. Pas faire ça. I'm sorry, I made something wrong again. Okay, that's now I'm going to conclude in the last 5 minutes in the role of how can we manipulate DC for immunotherapy. I'm not going to discuss the use of dendritic cell as vaccine. This is used since years in the field, and we will have, I think, a presentation from Dr. Barbuto on this approach. I'm rather going to discuss approach to manipulate, or how can we manipulate the DC in vivo. And again, there is all the different stages that are altered in the tumor environment by the tumor that can be manipulated. There is strategies that aim to increase dendritic cell development, and there is growth factor like FRIT3 ligand or GM-CSF that are used to induce dendritic cell development. GM-CSF is used for vaccine approach, combined to vaccine, and FRIT3 ligand is used directly in vivo in patients to increase DC numbers. There is also possibility to interfere with macrophage development that is at the expense of dendritic cell development, or if we block macrophage development through blocking CSF1 receptor, for example, we can increase dendritic cell development. Antigen uptake can also be increased. There is this don't eat me signal, CD47, SIRP-alpha. SIRP-alpha is expressed on macrophage, but it's also expressed on some dendritic cell population, except the CDC1. It's expressed on CDC2 in particular, and in PDC. So by blocking CD47 or SIRP-alpha, we can increase antigen uptake and favor antigen presentation. So this is one way to increase antitumor immunity. There is also strategy to activate, to restore dendritic cell function. So one key molecule is CD40. So there is agonist anti-CD40 that are currently in clinical trial to activate dendritic cells to restore their function. And there is other strategy to activate toll-like receptor, in particular for PDC toll-like receptor 7 and 9, or for CDC1 toll-like receptor 3. So there is currently ligands that are still in clinical evaluation. And we contribute in mouse models to develop several approaches based on TLR activation of dendritic cells using intratumoral injection. There is another pathway that is quite exciting currently, which is the SIGA-STING pathway. Although this is believed to occur in the tumor cell itself because this is linked to DNA damage, DNA that is cytosolic DNA that is sensed by the SIGAS pathway activate the STING pathway. But there is recent observation that the signal, the SIGAS signaling activation is occurring in tumor cell, but the ligand, the SIGAMP, can be sent in the extracellular milieu by the dendritic cells in the tumor environment. because tumor cells will be impaired for their capacity to respond to this pathway, but this activation can still be sensed by the dendritic cells in the environment. There is also an approach based on oncolytic viruses that will not only kill the tumor cells through lysis of the tumor, but also activate the immune system through the release of nucleic acid from viruses that will activate dendritic cells. And we had one recent contribution using a strain of rotavirus that will also induce tumor cell death and immune activation. DC can also have suppressive function. We already discussed ICOS ligands on PDC. So ICOS ligand, because external regulatory T cells can be seen as an immunosuppressive molecule expressed by dendritic cells. But also PD-L1 and PD-2, which is on tumor cells in some tumors, but in the vast majority of tumors, it's expressed by myeloid cells, dendritic cells, and not other myeloid cells. So it's likely that the impact of anti-PD-1 through anti-PD-L1 may interfere between dendritic cells and T cell activation to restore anti-tumor immunity and may contribute to the clinical activity of this molecule. There is other immunosuppressive molecules that are expressed or can be expressed on myeloid cell or dendritic cells, and we have a particular interest and focus on CD39 and CD73 that are involved, that are icononucleotidases that are involved in the production of adenosine, and adenosine is a potent immunosuppressive molecule that will block most immune cell function. And these molecules are currently targeted in clinical trial. And there is other pathways, other molecule, immunosuppressive molecule expressed by myeloid cells that will target essential amino acid in particular. And these are enzymes that are also targeted by small molecules and are also in clinical trial. I will stop here and just would like to mention the name of few people. So the work on PDC is the work of Nathalie Bendris. So this is the recent collaborators, current collaborators, the group of Nathalie for PDC in collaboration with Christine Ménétrier-Caux for the interaction with the regulatory T cells. The work on CDC1 is the work of the group of Jenny Valado. And we have a strong effort currently to analyze the spatial contexture of tumor cells. This is mainly through development of multi-immunofluorescence by the team of Bertrand Dubois. And this work is mainly based on access to tissue from patients. And this is allowed thanks to good collaboration interaction with the clinical department from the Centre Léon Bérard. And also, we'd like to thank the patients. And I would like to mention two collaborators, Daniel Olive for the work on ICOS and Marc Dallot for the work of CDC1 in mice. And I thank you for your attention, and I hope I will still have some type of questions. Thank you for your attention.
Thank you very much.
Very nice. It's really a class about disease. We learn a lot with your presentation. I think students are very happy. I don't know, Professor Barbuto, if you want to start.
No, go ahead. I'm having trouble with my computer here now.
Okay. Now, I have one question regarding this last images about the interactions of DC subsets in the tissue. And I was wondering if you can probably think about if patients presenting a high number of interactions between DCs1 and PDCs could probably have a better prognostic curves. Have you ever looked at that?
Yeah, so this tool is in place, but we still have some but it is time-consuming in terms of analysis for the moment, in terms of automatized analysis, and it's still not perfectly working, I would say, and in particular to analyze contact and interaction between and proximity between cells. Mm-hmm. So that's where we are in terms of analysis. In terms of making the staining, we can do quite large number, hundreds of patients, several hundred of patients is feasible. The step of analysis is more time consuming for the moment, for us at least. We are not at the, we'll still need some time for that. But clearly that's where we are moving to. It's really to not only to analyze one population or two populations, but their proximity and in terms of impact. And this may allow to reconciliate some observations that we had in the bad prognosis of PDC, and maybe in some situations they may not be of bad prognosis if they are in the right position.
Yeah, very nice. More questions here? Pierre, yes, please. We don't listen you.
Sorry, very nice talk, thank you. I wanted to ask you if you know, if you have some information on the T cells interacting with DC1 inside the...
Oh, that's also something so the way we have made the multi-IF, we had one multi-IF of 7 color for T cells, and it's linked with DC lamp, and one for the DC subset, but so far we have not linked the different DC subset with T cell functional activity. So PD1 or Ki67 or Caspase-3, for example. So that's the next step. We can do that, but we have not yet developed it. And what we expect to be able to do is to combine two multi-IF on cellular section. This is another technical aspect that I hope we can move. So I have no answer for that, but that's of course an important aspect. Yeah.
We have one question from people from YouTube. That's some while to...
Yeah, we have a question from Nadine Jimenez. She's asking, what's your opinion or experience on generation of immunogenic cell death of tumor cells to stimulate disease and initiate an immune response?
I have to say that we had some historical experience a few years ago, and while we were looking for cross-presentation, the readout was cross-presentation, but was not really DC activation. That was really a specific feature of DC, and we did not have the right tools or the right models to be convinced of the data, I would say. So I did not further investigate, but today there is we may be rediscovering other form of tumor cell death. Maybe apoptosis is not so immunogenic, but there is a pyroptosis which may be not necessarily triggered by caspase, but by gasdermin, that may lead to kind of inflammatory cell death that may be more prone to induce anti-tumor immunity.
Very nice. If we don't have further questions, we can probably move. I will thank very much, Christophe, for your time.
Thank you again for the invitation.
And yeah, we need to collaborate also. We need to continue this collaboration in the near future.
You know.
Thank you.
Thanks.
So I will introduce the next speaker. Dr. Pierre Guimond-Prez. Dr. Pierre Guimond-Prez was group leader and faculty at the King's College in London from 2012 to 2020. He's now director of the research at the Inflammation Research Center in Paris, France, directing the team of phagocytes and T-cell immunity. Dr. Guimond-Prez's research is mostly focused on DC, dendritic cell ontogeny, hematopoietic DC-based vaccines, and T-cell activation in the cancer context. His group have published key works lately understanding the features of a new subset of disease, the DC number 3, in the tumor context. So I'd like to thank you very much for your time. It's a pleasure for us.
Okay. Thank you very much for the nice introduction and for the invitation. So I'm delighted to participate to this very exciting workshop. So first I'm going to acknowledge my collaborators, sponsors, and my team members for fantastic collaboration. And I would like to especially thank Rodrigo for our previous collaboration when he used to be at the Curie Institute, and I hope we'll have new opportunities to keep collaborating. So I would like to split the talk in two parts. One first part, which is more a kind of a lecture, trying to review very basic concepts on dendritic cells, following the very nice introduction by Christophe from the previous talk. And a second talk, which is second part of the talk, which is addressing a very specific question that we are currently working on, which is the activation of CD8 T cell subsets. So what are dendritic cells? So dendritic cells, they are...
Sorry, Pierre. We cannot see your screen moving. Yeah, now yes.
You see?
Yeah, you can probably put in presentation mode.
You see here? No?
Not yet.
So this is full screen now.
I think for us is like an open PPT, but probably we can move.
But I think this is the fourth slide.
Yeah, this is the fourth slide. Yeah.
So you can see now.
Yeah, it's not in presentation mode, but we can see your slides. Yeah.
I can make it like that.
I think it's okay. Yeah.
Yeah. Sorry, I don't know why it's not. The full screen is not working, right? No.
No.
Okay. But anyway, I can go on like that, if you don't mind.
Okay.
So basically, dendritic cells they rise from hematopoietic stem cells, and they fuel the tumor immunity cycle because they can pick up antigens at tumor sites and then undergo this process of maturation, which is concomitant with two things: with migration toward lymph nodes and antigen presentation by MHC1 and MHC2 molecules. So upon activation and reaching the lymph nodes, the dendritic cells activate naive T cells and also innate cells like NK cells. And those effectors can, in some instances, reach tumor sites and fulfill effector function. So there is a lot of different kind of dendritic cells, and but all the key feature of dendritic cells, all the dendritic cells share some feature which are that they rise from specific progenitor in the bone marrow, and most of them respond to flat-free ligand and also to some extent to GM CSF. They migrate from tissues to lymph node, something that macrophages do not do, and they activate naive T cells. So looking in more details in the bone marrow, there is now recent work as a precise ontogenic pathway of human dendritic cells from hematopoietic stem cells. So dendritic cells sit a little bit in the hematopoietic tree between lymphocytes and monocytes. And you have different type of dendritic cells. So plasmacytoid dendritic cells, which have a lot of similarities with lymphocytes. And more myeloid, I would say, dendritic cells, the DC1, we heard about previously, DC2, and the recently described type 3 disease. I have to say that some people also call them DC2B. So they are very similar to the DC2s, but they share a lot of feature with monocytes. So they sit a little bit in between. And so this is a summary of the tentative summary of the current knowledge on the dendritic cells, excepting without the PDCs. So about the dendritic cell type 1 and type 2. So there is a strong level of conservation in the domain of the type 1 dendritic cells because they express some both murine and human express XAR1, for example, and also CLEC9A lectin. They express high level of IRF8, which is very important for their development. In terms of the type 2 and family of dendritic cells shares the expression of C11b and SIRP-alpha, for example, and IRF4. But in human, you have expression of CD1C and other additional monocyte-related molecule. In mice, there is a genetic control which is distinct between two type of type 2 dendritic cells, because both of them rely on IRF4, but some of them rely on Notch2, while other one rely on KLF4. And this is not known in human. So dendritic cells, a key feature is that they migrate from tissue to lymph node by afferent lymphatics. So it's a process that is affecting all dendritic cell subsets. So they reach the lymph nodes and they won't go further. It's like only activated T cell that can reach back the bloodstream by the lymph. So why do dendritic cells matter in cancer immunity? There is a lot of different evidences. I'm going to show you some of them recently published. One of them is, for example, a very nice study from the Krumel lab in which they show that the signature genes defining type 1 dendritic cells are associated to favorable outcome in melanoma and also lymphocytic infiltration. And it's also important for to predict the response to anti-PD-1 immunotherapy in melanoma. And so the more type 1 dendritic cells are found in melanoma, the better the response is. As you can see, responder display more level of infiltrating type 1 dendritic cells. And the genetic evidence come from the mouse system because the lab of Ken Murphy has produced some DC1 deficient mice, genetically deficient mice. And now more refined models are now available. But this first study has really shown that the rejection of immunogenic tumors is compromised in those DC1 deficient mice. Together with the activation of CD8 T cells. So there is a mechanistic link between the activation of CD8 T cells against tumor and type 1 dendritic cells. And regarding the response to immunotherapy, animal models have also provided genetic evidence that immunotherapy by checkpoint blockade is absolutely dependent on the presence of type 1 dendritic cells. So, for example, here in a study from the Myriam Merat's lab. So what is important also is that the infiltration of type 1 dendritic cells is very the intensity of the infiltration is very regulated by a lot of inflammatory chemokine, among them XCL1 and CCL5. So XCL1, which is engaging the DC1 specific XR1, and CCL5, which is engaging CCR5 chemokine receptor, which is expressed on a lot of different cells, including activated T cells. And those inflammatory cues are, some of them provided by NK cells or activated T cells, are very important to bring DC inside the tumor. So what are the key signals that activate dendritic cell in cancer? So it's a big topic, but I'm going to provide you some examples. And so it's important because, as Christophe nicely explained, so there is this process of maturation, and maturation is associated by the upregulation of membrane and soluble molecule, which controls the activation of T cells. So you have both activatory and inhibitory signals, and it's key to understand the function of dendritic cells to understand how they get activated and how they mature. And a prototypic activation of dendritic cells is achieved by activation of toll-like receptor, which drives the production of IL-12 in T lymphocytes into interferon gamma producing cells. And it's also true for NK cells. Another very important aspect is activation of dendritic cells by key innate signals derived from dying cells, and including tumor cells engaged in immunogenic cell death. And this is as described by the work of Guido, a lot of different cues that are released from dying cells, including ATP, HMGB1, or calreticulin that can drive the activation of a mature phenotype in dendritic cells, leading to the production of IL-1 beta, which is key for the activation of effector gamma delta and CD8 T cell responses. Another important aspect of dendritic cell activation is the ligation of CD40, as we have heard before again, which can be provided in many instances by CD40 cells that can drive the activation and license cross-presenting dendritic cells to better recruit and activate CD8 T cells. And here, so CD40 is a key effector, but you also have chemokines recruiting CD8 T cells and also cis interaction between T cells. And lastly, another last modality of dendritic cell activation, which is highly relevant for cancer immunotherapy, is the activation by T cells when they get reinvigorated by a PD-1 blockade. So the group of Michael Pittet has shown that this triggers a very efficient activation and very rapid activation of dendritic cells, which is dependent on the release of interferon gamma by activated T cells, which engage the interferon gamma receptor on dendritic cells, which in turn produce IL-12. And there is a positive cycle which drives and reinforces the activation of T cells. So again, another modality of dendritic cell activation. So now I'm going to focus more on of CD8 T cells. You have different kind of memory subsets. Basically, this can be divided in three main categories. You have the central memories, the effector memories, and the tissue-resident memories. So the central memories are characterized by the specific retention inside lymphoid organs, lymph node, by expression of CCR7 chemokine receptor and S1PR1 receptor as well. By contrast, effector memory cells, which are also called circulating memory cells, can leave the bloodstream to reach inflammatory sites, and from there they can migrate back to lymphoid organs and again recirculate in the bloodstream. And this is orchestrated by the lack of CCR7, but the expression of S1PR1, which enables egress from lymph nodes. And specific homeostatic receptor for homeostatic cytokine like IL-15, for example. So those memory cells, they can enter tumor sites potentially, but they can recirculate. And lastly, there is a very interesting type of memory cells, which are the tissue-resident memory cells. And those cells, by contrast to those cells, they do not express S1P receptor and this familiar receptor, and they tend to reside into these tissues. And they have a specific transcriptional program which is orchestrated by BLIMP1 and OBIT, most particularly transcription factor. And they express a specific set of adhesion molecules, including integrin CD103 and CD49, and also chemokine receptor associated to the entry and retention into tissues. So these cells are really programmed to stay inside tissues. And in the context of tumor, they display an activated phenotype characterized by the expression of CD103 and CD69, and low level of CCR7 and S1P receptor. And importantly, the group of Sherrin-Loy has shown that this is the infiltration of these TRMs, these tissue-resident memory cells inside breast cancer, more specifically in triple-negative breast cancer, is a better predictor than CD8 alone to predict favorable outcomes. So we have characterized dendritic cells subpopulation infiltrating luminal breast cancer, and we found that there is a very rare type 1 dendritic cells that we have seen before, but also some type 2 and type 3 dendritic cells. and the infiltration of the different subsets is uncoupled, is not correlated, except the infiltration of type 1 and type 2 dendritic cells. And by trying to characterize more specifically the functional capability of these type 3 dendritic cells, we realize that they are very good at activating the differentiation of T cells. All dendritic cells can activate T cells, but the type 3 and the type 1 to a lesser extent can drive to the acquisition of CD103 positive CD8 T cells. So, by contrast, where monocytes cannot and macrophage cannot drive the activation of naive T cells in vitro. So we look more carefully at the transcriptional landscape of those CD103 CD8 T cells activated by type 3 dendritic cells, and we found that they express a proper TRM signature, the same signature that you found in a triple negative breast cancer or TRMs infiltrating this type of tumors. On a limited number of samples, we could analyze the content of CD8 TRMs in luminal cancer, and we found that they actually correlate with the infiltration of type 3 dendritic cells, but also than with other subsets. So the role of TRM is not limited to breast cancer, and there is actually way more data in which the group of Fatia Mami-Chouaib and has done pioneer work, have refined the vision, the diversity of CD8 T cells in human lung cancer, and the study can distinguish between the OBIT expressing, so ZNF683 is the other name of OBIT, which is a key transcription factor for CD8 TRMs, and so this cluster of CD8 T cells is in fact associated to favorable outcome, and so we, even if they display a lot of phenotypic similarities with dysfunctional T cells expressing, for example, the LIM marker, they do not have the same predictive value in non-small in NSCLC. And more recently, the group of Fatia Mami-Chouaib has also shown that the accumulation of CD103 cells is a predictor of clinical response to a checkpoint blockade. So we were interested to better understand the activation of these TRMs in the context of lung cancer. So we came back to mice, and we used an endogenous model of lung cancer by which is overexpression of a mutated form of KRAS, and that can be derived as an transplanted model. And we have seen that in both endogenous or transplanted model, we have a nice infiltration of tumor sites of TRMs that express both CD69 and CD103. And those TRMs express a dysfunctional phenotype with expression of multiple co-inhibitory receptors like TIM3 and PD1, for example. So from the myeloid side, those tumors are infiltrated by a major neutrophilic infiltrate, but there is also an accumulation of type 1 and type 2, I will say, for all CD11B expressing DCs. In more detail, the type 2 DCs change their phenotype when the KP tumor develop. And we found this CD11B DC that express a lot of CD103 and the CLEC10 lectin. So there is a change and an inflammatory recruitment, which is orchestrated by GM-CSF derived from the tumor. So in the first attempt to try to understand the role of dendritic cells, so what we have done is to use the dendritic cells deficient mice and compare them to monocyte deficient mice. And we found that inactivation of all dendritic cells compromise the development of TRMs inside the tumors. And this is not the case for monocyte deficient mice. Then we decided to look at in more details what's going on in the mediastinal lymph node, the lymph node that drains the tumor mass in the lungs. And we found that both type 1 and type 2 fluorescent antigens expressed in tumor cells. So they can bring tumor antigen inside the lymph node. The type 1 are more efficient than the type 2, which was a little bit expected given the literature given the fact that they have a higher capability to phagocytose dead cells. And when the dendritic cells are sorted out from the mediastinal lymph node, they can activate CD8 T cells specific for the tumor ex vivo. And the migratory DC do it much better than the resident DC. So when we look in more details and we compare the migratory DC of type 1 and type 2 phenotypes, we saw that the CD8 T cells, but those CD8 T cells express very little TRM markers. By contrast, type 2 dendritic cells are very poor at activators because in part because they uptake less antigen, and those CD8 T cells undergo efficient CD103 expression. And adding back IL-12, which is a feature of DC1, IL-12 production inhibits the expression of CD103. So those two types of dendritic cells have a distinct capability in driving the activation of TRM. So in more details, we try to restore more antigen inside the DC2 by adding MHC class 1 binding peptide on the cells, and we saw that if we add increasing amounts of peptide, we can restore a very high level of proliferation by DC2. So if we add more antigen, so showing that the DC2 were limited to present to CD8 T cells because they cross-present poorly. But by contrast, we see that despite the strong proliferation, we lose the acquisition of CD103 in activated cells. So we think that the activation of TRM is biased toward inefficient cross-presentation. And finally, we decided to test which cells was required in vivo. So we developed, we use in collaboration with Marc Dallot this XR1 DC1 deleter mice, and we compare them to IRF4 deficient mice only in dendritic cells. So as control, we lose specifically the DC1 in this model, and we lose specifically the migratory DC2 in this model. And the outcome is that when those mice are engrafted with these KP lung tumors, we could found a specific inactivation of T effectors. So first, in TRM, sorry, I'm starting with TRM, it's going to be easier. So here we have this TRM in the control. So they get completely removed when DC1 are absent, but also when DC2 are absent. So it was very surprising, but apparently we found that you need both DC types to obtain this TRM response. By contrast, when we look at other activated T cells that are not TRMs, but express high level of CD44, we see a specific effect for DC1 deleter mice. So those effector response are largely independent from type 2 dendritic cells. So these are very important. So we are working on the mechanism of this finding, but we think that this has important implication for immunotherapies, because if you want to activate TRM response, you might have to use both subsets of dendritic cells. So here is the graphical summary of the function of dendritic cells in more, and I will be happy to take some questions.
Very nice, Pierre.
Thank you.
I think we have already some
expression of chimeric receptor in myeloid cells, but I think most people are pursuing monocytes and macrophages. But I think you're right. There is a strong rationale to also try in type 1 dendritic cells, because they might expand the response by priming efficiently CD8 T cells. However, the problem is that technically it's probably quite difficult because you need to expand those cells and to get them proliferating, which is probably not easy, but I think the rationale is good. So why not try?
Okay, thank you very much.
Very nice. I can probably, Christophe, Christophe, you could go ahead, please.
Thank you, Pierre. So I have just one little small question regarding the expansion or induction of TRM leading the cooperation between the two cell types. So you mentioned that's maybe linked to a lower level of T cell activation, engagement of the TCR, I don't know. But from my understanding is that there is a need of TGF-beta to induce TRM phenotype, or maybe it's not needed at the priming step. It's not clear to me. But do you think it's linked with somewhere with TGF-beta and the capacity of one DC to produce TGF-beta and not the other, or maybe to cooperate for the activation of TGF-beta?
Yeah, so thank you for asking, because it's a very interesting question, because there is a group of memple who has shown that if you remove TGF-beta presentation from all DCs, you lose TRM activation. But what they think is that this is an effect that is achieved on naive T cells. So the DC, they prepare the naive T cells to become TRM via TGF-beta presentation. That's their model, if I understood well. Okay. And what we found is that actually the naive T cells from DC1 deficient mice, they have decreased the CD103 expression. So we think that the DC1, and this is not the case when we remove the type 2 dendritic cells. So we think that the DC1, they have a function of, they have an interaction with naive CD8 T cells that might be dependent on TGF-beta. This we don't know yet. And this is preparing the naive CD8 T cells to, this is poisoning the naive CD8 T cells to become TRM.
Okay, so you think that the source of TGF-beta will be the CDC1? That your hypothesis?
Yes, but
or the activation.
This might be partly before the development of the response.
Yeah.
This might be achieved by an effect on naive T cells and not by presenting cells.
Okay, thank you.
Yeah, but we need to do more genetic experiments with DC1 specific inactivation of TGF-beta that we haven't done yet.
Okay, thank you, Pierre.
Thank you.
I probably have one little, very fast question regarding the TRM in patients. Have you ever, I don't know, in the literature probably, it was shown if the DC subsets and TRM CD8 cells are in contact in tissues and patients and different lymphoid organs, or even, I don't know, bone marrow?
No, what has been shown, I think, to my knowledge, what has been shown is that in viral infection, in herpes infection, cervical infection, you have an interaction between this inflammatory type 2 dendritic cells, like type 3. It's not clear exactly what they are in mice, and TRMs in tissues. And now there's a recent paper that just came out, which is showing in cells that there is CXCL16, which is a ligand of CXCR6, okay, which is a chemokine receptor in TRMs, and which is important. So you have mature dendritic cells inside tissues, which would be interesting with TRMs as well, a bit, without migration. So, but not specifically type 1, actually.
Okay, thanks. More questions? I don't know. In YouTube, probably not. So we need to move forward. Thank you very much, Pierre.
Okay.
And see you soon also, probably in Brazil. Okay. So I introduce the next speaker. It's also an honor for me, for sure, to introduce Professor Barbuto. So Professor Barbuto is medical doctor and professor associated to the Department of Immunology and to the School of Medicine, both from the University of São Paulo. Professor Barbuto has dedicated his research to cancer immunotherapy, mostly focused on the study of DC-based therapies for cancer patients. Professor Barbuto has supervised more than 70 students among master, PhD, postdocs, and he is a pioneer scientist in the cancer immunotherapy field in Brazil. So everybody likes Professor Barbuto in Brazil. So thank you very much, Professor.
You missed the main part. I was your supervisor from very early. and I think actually I'm spending money, but that's a sign that I did something good. My students are becoming great scientists. So that's a good thing. Okay, let's move
Very
on.
nice. Thanks.
Am I presenting?
Yes.
Okay. So, well, it's very easy to present this. Normally I spend a lot of time setting the context, but in this case, after all these fantastic talks, this context, the immunological context is perfectly explained. I'll present some preliminary data on a clinical study we're doing with dendritic cell vaccines in patients with glioblastoma. What we don't have the context is the context of the disease. Glioblastoma is a brain tumor. Actually, among the brain tumors, it's the most frequent, and it's very aggressive. Before the introduction of radiotherapy, the survival, less than 50% of patients survived for 6 months after diagnosis. With the introduction of radiotherapy, and that was almost 40 years ago, the survival at 6 months increased, but if you see at 2 years, almost between 90 and 80% of the patients were already dead. And despite of continuous research in the area, not much has changed. If you see, actually, education keeps changing. But if you see the survival here, the survival curve for patients with glioblastoma is still very poor, and that was 5-6 years ago, and it's still very poor. The only relatively good thing is that in Brazil we do get similar results. If you look at the survival for the patients, it's among the median survival is still around 15 months, which is worrisome. If you have, and this disease gets very frequently young people, so it needs new forms of therapy. And since immunotherapy became a reality, immunotherapy for glioblastoma began to be searched with very intense efforts. And among the strategies are the dendritic cell vaccines. We don't have to talk about dendritic cells, but I think this picture is very nice. It illustrates what dendritic cells seem to do. Actually, they pick the right cells to present, considering that they are good. The dendritic cells are working well. So very early, as soon as it became possible to generate dendritic cells, cancer was cured, in theory. You should generate dendritic cells from the patient, and that became possible when you learn how to differentiate monocytes into dendritic cells. You would prime these dendritic cells with antigens and re-inject the dendritic cells back into the patients, and the tumors would be controlled by cytotoxic T lymphocytes. Obviously, that's not so simple. As we learn more about dendritic cells, as we did today, you realize that there are dendritic cells and dendritic cells. It's not easy to generate the dendritic cell that will generate the effector cell you want. But even worse than that, and this is Rodrigo's work. If you take dendritic cells from patients, monocytes from patients to generate dendritic cells, you get very frequently dendritic cells that are not good inducers of effector cells. Actually, they prefer to induce regulatory cells that will produce a lot of IL-10, a lot of TGF-beta, and no interferon gamma. So you have, in theory, a strategy, but not so easy to put it into practice because there are many unknowns. One strategy we have been studying is to use...
Sorry, Professor. I think the presentation is freeze in the slide about DC-based cancer vaccines.
Wow. So yeah. I'll get out of it and go back. Cancer base. This one?
Yeah.
Did it move forward now?
Yeah.
Okay. So, well, just I've shown that dendritic cells generated from blood monocytes from, in this case, breast cancer patients, have a preference to induce regulatory T cells that express FOXP3, secrete IL-10, secrete TGF-beta, and do not secrete interferon gamma. So an alternative strategy that we have been trying is to generate dendritic cells from healthy donors and to obtain the tumor from the patient and fuse them. Actually, you have to fuse. You cannot just obtain the antigens here because these dendritic cells are not histocompatible with the cancer patient. But by doing that, you generate hybrid cells that express potentially the antigen-presenting ability of dendritic cells and the tumor antigens, and you get a bonus effect, the allogeneic effect. That was shown many years ago, lots of years ago, that when you mix any antigen with allogeneic cells, even better if they are dendritic cells, and inject that, you get a much stronger response. Presumably because the allogeneic cells will activate more cells and enhance the cytokine environment to induce a response. So that's the strategy. The first study we published in 2004 and showed that you do have a response. In that case, were patients with melanoma and renal cell carcinoma. And after you generate the hybrid cells, you would inject with intervals between 1 and 2 months after irradiating the cells. So you just have the stimulus but no proliferation of cells in the patients. In glioblastoma patients, when they decided, we've been trying to do that for a long time, but now we recently started a study. Patients were recurrent GBM for glioblastoma. That were resubmitted to surgery and then followed the standard protocol, chemo and radiotherapy, mixed with a vaccination. The tumor samples were processed and kept frozen, and for each dose, you defrost the cells and fuse them with dendritic cells generated from healthy donors. Patients were followed clinically as standard, and in the immunological follow-up, we analyzed the tumor-specific circulating CD8 and CD4 T cells. This is a very preliminary result, mainly because we had there was a change in classification. Now glioblastomas were split in two different tumors, astrocytomas grade 4 and glioblastoma, and this mixes everybody. But if you compare that with a median survival of patients treated at the same institution they were not paired, it's just a historical control without vaccine. And the median survival we've been obtaining with the vaccine, it does seem to make a difference. The question is, how is it working? And for this, I still do not have the answer. We are still analyzing the data, but I will just present the results from one case that's been submitted for publication. It was a patient with 20-year-old female patient previously healthy, that appeared in 2014 with an anaplastic astrocytoma, which after treatment recurred 3 years later, and when it recurred as a glioblastoma. In March, 3 months later, she was reoperated and treated with radiation again. 3 months later, the tumor relapsed, and there was no treatment available for her. So she was the first patient actually enrolled in the study. And the clinical results were impressive. She had a very ugly tumor. It's easier to see in this series, a very large tumor. But already after the first dose, you had some decrease of the size of the tumor, which actually improved a lot.
Sorry, Professor, we cannot see again. It's through. Yeah, now okay, but there is a delay in the.
And now?
Now it's okay. Now we can see the picture. Okay.
So you see before the treatment, after the recurrence, the relapse in August, she had a tumor that almost filled the right hemisphere, the right brain hemisphere. But it decreased very quickly already after the dose, and it kept decreasing, kept stable up to the 6th dose. Then she started at the 10th dose, 10 months, actually it was monthly injections. She has shown some progression, and by 1 year, the tumor had progressed tremendously. All these scales of quality of life and the tumor, actually, you had, after the 2nd dose and radiological evaluation of the tumor, you had partial response that remained as that, and then started to stable disease and progressed. And if you look at all this, you have a clinical stability or improvement of quality of life up to the 10 months of treatment. If you look, but when it progressed very quickly, and she passed away 3 months after stopping the treatment. The curious thing is that this patient had she had no circulating tumor-specific T cells, neither CD4 nor CD8. Actually, she had cells circulating, but you had to expand them. And when we did that, we cultured the circulating T cells with the tumor. After 5 days, we stimulated with IL-2 and actually evaluated the proliferation, sorted the positive cells, expanded them in vitro, and then it was possible to analyze what she had. She did have T cells of very low frequency that was not detectable in this simple proliferation, initial proliferation assay, but she had, and you see she had quite a few Treg cells secreting IL-10, which after the 2 doses already decreased considerably and stayed low till the 6th dose. And then they started to come back, and at 9 doses she had already, but never as much as she had in the beginning. You looked at other cell populations, and you see she had.
Sorry, Professor. Is still, yeah. Now it's okay. Now we can see interferon gamma, IL-17.
Yes.
Okay.
It was a brief presentation, so I engineered this way so it would take longer. So she had a Th1 cells, interferon gamma secreting cells circulating. It was a very low frequency, below the detection level in that assay, but she did have. But these cells increased, and not so much in the 2nd dose, but increased and increased. And actually, if it's better to look at the lower graph here. Before treatment, she had a lot of cells. Th1 cells were predominant cells, but she also had Th2, Th1/2, Th17 ones. It was very heterogeneous population. With the vaccination, the Th1 cells predominated very clearly up to the third dose. But this changed dramatically, and it started to change already at the sixth dose. I'm not showing here the summary because this is, but it started to change already at the sixth dose, where Th17 cells increased very clearly, almost none here, and increased very clearly, and also Th1-Th17. At the seventh dose, what increased was Th2. And then Th17 predominated and was with that for a long time. Basically showing that the vaccine was able to induce a Th1 response, increase the frequency of T cells responding to the tumor. But after some time, this response was deviated and it went to other circulating CD4 T cells, suggesting that maybe these cells have a role. The subpopulation and Th17 cells in this patient do seem to have been a very negative, that have a very negative effect. The other interesting thing to look at here is that the main response we saw in the blood was CD4 T cells. CD8 T cells were never as frequent, and they varied very little throughout the course of the disease. It could be that these CD8 T cells were within the tumor. That's why we didn't find them in the circulation. But I think we should look more carefully at CD4 T cells, at least if we're looking at circulating T cells. And now just to show the people involved in the study, in the Laboratory of Tumor Immunology and LIM 31, all these people. Patricia is the main scientist involved in the vaccine preparation. Mariana had a lot to work in the characterization of the T cell populations. And all the other people, all the other students actually worked a lot because all these analyses take a lot of time. In the neurosurgery and clinical follow-up, the participation of Dr. Lepsky was essential. We've been trying to do this to the study in GBM for a long time, but we could never find a partner that took it ahead, and Dr. Lepsky took it ahead and actually generated all this data. Dr. Venancio was the one that studied the slides in pathology. And thank you very much.
Thank you. Very interesting data. I think Christophe has a question.
Yes, thank you, Alexandre. I have one question. So you fuse the tumor cells and the allogenic dendritic cells. So yes. I don't know, glioblastoma, are they expressing class 2 as well?
Beg your pardon, I didn't...
Are they expressing the MHC class 2?
Yes, they do. Actually,
okay.
They do. Actually, the fusion process enhances the expression of MHC, both class 1 and class 2.
From the tumor itself. Okay. Because this is probably key, because you bring the endogenous CMH, or class 1, it will be expressed in most tumors, but class 2, maybe not.
Yeah.
Okay. In glioblastoma, it will come with the tumor. Okay, that was my question. And that's fit with the fact that you are seeing CD4 mainly in the patient. And I have another question. How are you measuring the tumor-specific response? Is it ELISpot? Is it?
It's basically by proliferation. Actually, we present the tumor cells with the monocytes. Actually, we pulse dendritic cells, in this case, from the patient with the tumor antigen and then present to the T cells.
And then how do you prepare the tumor antigen? Is it lysing?
Oh, yes, by lysing, freeze and thaw.
Okay. Thank you.
I've been trying to avoid choosing specific antigens. I let the dendritic cells choose the antigens.
I like the approach that because when we select neoantigens, it's linked to mutation, but that's what we know. That's what we can identify. identified, but probably there is other immunogenicity within the tumor.
Yeah.
I have one little question also in the same idea. If you still have material from this patient, probably have you thought to sequence the tumor from the patients during the treatment, and probably also check the clonality of T cells during the treatment?
Yeah, the T cells we can do. With the tumors we can't, because you cannot reach the brain every time. Some genes are already measured normally for the classification of GBM, but that's it. Yes, Christophe. You raised your hand.
I have another question that's for you, or maybe Pierre may have his own idea. I understood from the literature, at least from mice models, that the vaccine using dendritic cells, the dendritic cell is not the cell presenting the antigen itself, but there is transfer of the antigen onto endogenous dendritic cell. I don't know how comfortable you are with that vision. Do you think it's really what's happening in patient?
I don't know. To be specific. I hope it's not, because if the patient's dendritic cells have a bias, I would prefer the hybrid cells to present, but it's just hope. I don't know. And this vaccine irradiated cells, so I don't know how much they can transfer, but that's only suppositions.
It may be easier to transfer than to directly present.
Yeah.
Mm-hmm. I don't know.
Yeah, it's something to be tested. Yeah.
No, you don't work with mouse model, or you can?
I haven't been working for a long time, but I will go back to mouse models now. I think we've got stuck here with this, so we have to change.
Okay, so we need to go forward because we are short in time. So thank you, Professor Barbuto, again for your time, for the nice presentation. I will introduce the next speaker. He's from the new generation, I'd say. So Diego Martins is graduated in biomedicine with master degree in genetics and molecular biology. Currently, he develops his PhD thesis focused on in vitro and in vivo models in acute myeloid leukemia at Faculty of Medicine from Ribeirão Preto. And at this moment, he's doing his abroad PhD stage at the University Medical Center in Groningen in Netherlands. You talk about macrophages. Thank you very much, Diego.
Thank you, Rodrigo. Can you hear me?
Yeah.
Yeah, great. Let me share my screen. Which I believe you can see now.
Yes.
Great. So, good.
Very nice.
Very good. So thank you very much, Rodrigo. I would like to thank you, Professor Barbuto and Professor Vanderson, for the invitation. And today I'm going to talk mostly about an intriguing observation that we had a couple of years ago, and we have been working on it. So this project that is entitled the IM2 polarized macrophage microenvironment drives leukemogenesis and poor prognosis in acute myeloid leukemia was a project that we started at the University of São Paulo, and the results that I'm going to present were mostly generated by this great PhD student, Isabel van Rocha. And this project was a collaboration, as you mentioned, between the University of São Paulo and the University of Groningen in the Netherlands. And Professor Eduardo Rego and Professor Jan Jacob are the main supervisors. So it's, I think, needless to mention the importance of the tumor microenvironment in solid tumors, as we had this great talk from Professor Jérôme about the immune score and the immune component. So as we can see in this review, if we look for the contribution of the different immune cells in the prognosis of different solid tumors, we can see that actually they have quite a dispersed profile, being, for instance, some M1 associated macrophage more associated with good prognosis, and the M2 macrophage seems to be associated with poor prognosis in most of the solid tumors. But what caught our attention, actually working mostly with hematological malignancy, is the fact that the studies in AML particularly are very little about the contribution of M2-like macrophage in the prognosis of the patients. And if we just make a fun exercise to look for the expression of some M2 markers, and here I brought the CD163, using this cohort that is well established from the TCGA study, we can see that actually high expression of the CD163 is associated with poor prognosis in AML patients. And we wonder if actually what we are seeing here is the expression, because this is RNA sequence bulk, so we just sequence the mononuclear fraction, which includes the blast and the tumor microenvironment. So we wonder if this expression that we are detecting here, this is coming from mainly from the tumor cells, or this is coming from the tumor microenvironment that was sequenced together. So to try to address this question, we started by elaborating a FACS panel, whereby using the CD45, we could already distinguish mainly three populations. So we can see here a CD45 team, the leukemic blast cells, CD45 high with low SSC, or the majority are lymphoid cells. And we have these myeloid-associated cells that we could identify. And using a couple of markers that we all know that are expressed in monocytes, macrophages, and also in dendritic cells, we could already start to make some distinctions about the different types of monocytes, macrophage presence there, so classical, non-classical. And inside these populations, we used a couple of markers like the CD206 and the CD163 to define some sort of M2-like macrophage, and we used the CD8 to define the M1-like macrophage. And as we can see, the first thing that caught our attention is actually the fact that looking for the expression of those markers, they are mainly coming from this myeloid-associated component, but not from the leukemic blast population. So this actually caught our attention for the fact that most of these RNA sequence data that we are seeing out there, they are completely contaminated by this tumor microenvironment. Because if we now quantify the frequency of these cells in the samples, you will see that actually the expression is coming mainly from this myeloid-associated component. So to try to address that, we also took advantage from these digital cytometry techniques, such as the CyberSort that was developed by the Newman a couple of years ago. And we adapted the signature because the signature was mainly developed for solid tumors, where you can nicely distinguish immune cells and the tumor cells. And in the context of AML, since the cell of origin is a myeloid cell, this can be a bit challenging. So we adapted some single RNA data sets to try to purify the mainly contribution from the genetic signature from the blast cells. And we could also identify here the stromal component, the myeloid component, and the lymphoid component. And as we can see, taking advantage of one data set that we published a couple of years ago, whereby we sorted the CD34 positive, which would mainly represent the tumor cells, and the CD34 negative, which will mainly represent the tumor-associated microenvironment in that particular case. We can see that actually the contribution from the myeloid component and the tumor microenvironment is being detected by the CD34 negative compartment. So showing that actually the level of heterogeneity in infiltration of this immune component is quite drastic in these samples, where we always assume that we have a high component of tumor cells that are being sequenced. To try to address this point and to see the role of M2 macrophage in AML patients, we screened almost 70 AML patients by FACS, and we could already see that actually the level of M2 macrophage was associated with a more adverse and intermediate risk stratification using the European Leukemia Net risk stratification, and was also associated with one particular mutation in the FLT3 gene that was highly correlated with the frequency of M2 macrophage. We can also see that M1 macrophages are being associated with a more favorable and intermediate risk stratification. And if we now use the cybersort and try to identify the levels of M2 macrophage in some of these public data sets that are already published, we can correlate the drug sensitivity for several drugs used in cancer with the levels of M2 macrophage in these samples. And you can see that actually this strategy could allow us to identify some compounds that could be useful to treat AML patients with high infiltration of this M2 macrophage. So using this FACS dataset that we generated, we clusterized the AML patients, and you could identify mainly 4 clusters, whereby you can see that clusters 1, 2, and 3, there are clusters with high levels of M2 macrophage, and the cluster 4 is mainly categorized by a heterogeneous group of patients. And if we now look for the survival of these different clusters, whereby we take cluster 1 and 4 versus cluster 2 and 3, we can see that actually patients with the high M2 macrophage component, they are associated with poor overall survival and disease-free survival. And using the cybersort technique, we can see that actually, although we do not see between diagnosis and relapse a pattern between the levels of M2 macrophage being up or down regulated, we can actually see that the levels of M1 macrophage, they decrease in the samples upon relapse in these patients, suggesting that actually the evaluation of this immune component, it is important for the prognostic of these patients. So at the in vitro level, we could see that the co-culture of AML blasts with these different macrophage subtypes and using a stromal cell as a control could provide increased proliferation in vitro, decreased apoptosis induction, and increased drug sensitivity, drug resistance upon the main drugs used nowadays in AML clinics. And this was interesting to see that the co-culture of AML blasts with M1s and M2 macrophage was actually responsible for reprogramming this M1 macrophage into M2-like phenotype, whereby we could see the decrease in expression of CD8 and increased expression in CD163 and CD206 upon AML co-culture. So we decided to study the role of M2 macrophage in vivo, and to do so, we decided to make a PDX model, a patient-derived xenograft model, and we chose one very difficult subtype of leukemia. So in general, PDX models in AML, they are quite common and quite simple, but we know that AMLs with favorable prognosis, they are very difficult to engraft. And particularly, this subtype of AML, the acute promyelocytic leukemia, is well known for its difficult to engraft. So we decided to make a model whereby we isolated peripheral blood-derived monocytes, we differentiated them in vitro into M2 macrophage, or we kept them unpolarized in M0 state, and we transplanted intrafemorally this macrophage being the M2 in one side of the mice, in the right tibia, and the M0 in the left tibia, or as a control, we just injected saline solution, and we transplanted the APL blast via the retroorbital vein. And as we can see, measuring the levels of tumor cells in the control mice, we could barely detect any engraftment there. But when you look now for the presence of these tumor cells in the M0 legs, in this case in the left leg, and in the M2, we could see first that both presented high levels of engraftment there. That was not the case for the control. And you could also see that actually the leg with the M2 macrophage presented higher levels of engraftment than the M0 leg. And we also measured the levels of engraftment in the spine of these mice, and you could see that actually these levels, they are intermediate between the M0 and the M2 macrophage, suggesting that actually these cells after encounter the macrophage, they're able to migrate to other tissues and infiltrate the whole animal. We could also see that actually spleen weight was increased and the mice evolved with full-blown leukemia. And since we detected that the co-culture, in this case inside the tibia of the mice, between the tumor cells and the macrophage was able to promote the development of the leukemia, we decided to make this co-culture in vitro before and transplant the cells after they encounter the macrophage. So what we did, we again took advantage of peripheral blood-derived monocytes. We differentiated them in M0s and M2s, and we now took the APL blasts, co-cultured them for 48 hours, and we transplant them via the heteroorbital vein in immunosuppressed mice. And as you can see, the mice that received the APL, the blast cells alone, they never evolved with leukemia, so we sacrificed them after 24 weeks. Mice that were received the blast cells co-culture in M0 macrophage, they had a survival span between 14 and 24 weeks. And mice that received the blast co-culture M2 macrophage, they had a lifespan between 10 and 16 weeks. So we also, after the first transplant, we sorted back the tumor cells and the transplant in a limiting dilution fashion these tumor cells to evaluate if they are able to engraft in secondary transplants. So as you can see, this is a result from the first transplant. Mice that received the APL alone, the blast cells alone, they never evolved with leukostosis. They remained without engraftment through the whole period of the transplant. Mice that received the M0, they had at week 12 on increasing leukocyte count, and they evolved with full-blown leukemia being sacrificed at the end. But mice that received the tumor cells co-culture M2 macrophage, they actually evolved quite early with leukemia, displaying here signs of leukostosis and levels of engraftment quite expressive. And actually these mice, they are sacrificed around week 16, 12 to 16, to get the cells back to make the secondary transplant. And you could see that upon the sacrifice, the spleen weight was increased, and it's quite different if we compare the M2 macrophage, actually with even the M0 macrophage there. So you can see that the survival was different in the primary transplant, and upon the secondary transplant, this was even more striking. So animals that received 500,000 cells, they all had full-blown leukemia and they're being terminated. And mice that received 1.5 million cells, they had very short lifespan, which was never reported before for xenograft models in APL. So we decided to investigate mechanistically what was happening upon this co-culture. So we decided to repeat this co-culture of tumor cells in M2 macrophage, and we also took it along the stromal cells, in this case these MS5 cells, as a control of the cultural conditions. And you could see that actually comparing M2 macrophage with diagnosed, we have several genes that are upregulated and are only regulated up on the macrophage co-culture. And if we run some gene set enrichment analysis and GEO, we get actually change in metabolism, being these cells more OXPHOS driven, and also increasing cell proliferation. One of the first questions that we had was, is the encountering of M2 macrophage with the blast cells promote some sort of immune evasion in these cells? So we set it up in vitro phagocytosis assay, whereby we expose the tumor cells to M2 macrophage or to MSCs. And after this co-culture, we re-challenge these tumor cells with a new set of macrophage. And you could see that actually after the first challenging with the macrophage, tumor cells that received that experienced the co-culture, they displayed decreased phagocytosis in vitro. In a second challenge, we could not link this to upregulation of the main described don't eat me signals, such as the CD47 and CD24, but we could see decrease in these eat me signals, such as calreticulin, on the surface of these tumor cells. And this was also correlated with increasing expression of the stannocalcin 1 gene that has been recently reported to be upregulated in solid tumors associated with retention of carotecholine into the mitochondria, blocking the expression of the surface. So one of the first mechanisms that we observed was actually, yes, the challenging of tumor cells to M2 macrophage can enhance the escape of phagocytose and somehow could contribute to an immune evasion in these PDX models. A second question that we had, since some of the gene ontology terms associated with locomotion and migration, we asked ourselves if these tumor cells, they actually present a better capacity of homing after the transplant. So we performed an in vivo homing assay whereby we transplant tumor cells co-culture with M2 macrophage OR1 into the mice, and we sacrifice these mice after 16 to 18 hours, and we evaluate how many cells made it into the bone marrow, and you could see that cells that experienced this co-culture, they presented increased in vivo homing. This was also validated by transwell assays in vitro, and you could link this to increase the expression of some of the integrins that are associated with better homing in leukemia, such as the CD49D, E, and CD49F. And finally, one of the mechanisms that we observed was also upregulation towards a metabolism more dependent on the mitochondrial respiration. So we decided to first assess if this change in respiration capacity they were indeed true. So we performed a co-culture, and after that we sorted the tumor cells and performed a functional respiration assay by seahorse. And you could see here that in red are the tumor cells that were co-cultured in macrophage, in blue the co-culture with MSC cells. And you can see that actually these cells after the co-culture, they present increased basal respiration capacity and maximum respiration capacity. And that was also correlated with increased mitochondrial ATP production rate. And the interesting finding was that it's described that stromal cells, such as MSCs, they can transfer mitochondria to the blast cells. But the description for macrophage is still in the beginning. So we have a couple of reports that macrophage can do that, but not in leukemia particularly. But using labeled mitochondria in the macrophage, we could see that actually after the co-culture, this macrophage, they could transfer more mitochondria to these tumor cells than the stromal to the tumor cells, than the stromal cells. So just to address that and to try to translate these findings into the clinics, we know that actually the fatty acid oxidation is an important metabolic process for this M2 macrophage. And this has been described that actually the etomoxy treatment, that is a CTP1 inhibitor, is associated to reverting this M2 phenotype. So we decide to treat this macrophage or the stromal cells with etomoxy for 24 hours. Then we washed the cells and we performed the co-culture with tumor cells to see if now the cells, they would keep the same respiration capacity as we observed before, or if this was impaired. And we also combined that with one of the main drugs that is used for AML, that is venetoclax. It's a BCL2 inhibitor. So you can see that actually the monotherapy etomoxy or the combined therapy with venetoclax resulted in decreased oxygen consumption rate in tumor cells after the co-culture with treated macrophage. But this was not the case actually for MS5 cells. So suggesting that actually fatty acid oxidation can be one of the mechanisms whereby the tumor cells benefit for this co-culture and they change their metabolism to a more oxalose-driven metabolism. And finally, we could see that actually treating macrophage with etomoxy or the combination can revert in long-term co-cultures the phenotypes that we observed before regarding cell proliferation. So M2 macrophage, tumor cells treated with co-culture with M2 macrophage, they can proliferate for a long time without any support. But when you treat now M2 macrophage with etomoxy or etomoxy plus venetoclax, we actually reduce this proliferation capacity in comparison with the vehicle one. So far, we're still working, trying to understand which mechanisms are these tumor cells using to first have this interaction with the macrophage and what they have learn after this encountering with the macrophage. But we can say that actually tumor-associated macrophage, or M2-like macrophage, they provided growth and survival advantage to these blast cells, that the reprogramming of this M2-like macrophage would be very interesting to be incorporated into AML, because if we can revert these phenotypes, we could actually improve treatment strategies for these patients. And it's very important to make the distinction between the tumor cells and the macrophage populations to make proper conclusions about what we are actually evaluating when we use these different markers of survival in the patients. So I would like to acknowledge the groups that were involved. So this was a mood-centric study. And as I mentioned, this was one project that was developed by this PhD student, Isabel and Rocha. And I would like to address the questions, if any, and thank you very much for your attention.
Thank you, Diego. Very, very nice presentation. Very complete story. You go description and mechanism. I don't know if you have questions in the audience. We can have.
I think we have time for one question.
Yeah.
Because we already surpassed the time we had.
I can probably do this question. Do you have some clues about the mechanism? So what kind of cytokines or surface molecules on your M2 macrophages can give rise to this kind of tumor cell maintenance? There is a MSSF dependence, or there is other kind of cytokine?
That's an excellent question. We are actually running this experiment right now. We're labeling all the proteins on the surface of this macrophage to do a pull-out after the co-culture on the tumor cells and see how we can capture this exchange or this difference. But so far, I cannot give you a proper answer to that. Okay. But we're working on it.
Work in progress. Work in progress.
Yes.
So let me introduce the last speaker, which is a pleasure. As I said, Rodrigo was my student. We met in 2009. And I won't do a formal presentation, but he told me to do it, but I won't do it. We met in 2009. He came to study as a master's, and he studied till his PhD, which he received in 2015. But during his PhD also, the work for his PhD, he already worked in Dr. Caux laboratory in Lyon. So his PhD is both Brazilian and French. And then after receiving his PhD in Brazil, he went to France and spent 5 years there, right? As a postdoc scientist. And then Vanderson went to France and brought him to Brazil, which was a great thing for Brazil. And he is the one that really organized this meeting, so he deserves congratulations. Thank you, Rodrigo, and please.
Thank you. Thank you, Professor. Do you see?
Yes.
Okay. Sorry. Okay. So I'd like to thank you, everybody, for still being now here, because it's time for lunch for us. I try to be fast. So this work was developed at the time I was in Institut Curie in Paris. And we are trying to publish a history about a new subtype of macrophage called folio 2 macrophages. And we applied single cell RNA-seq and other techniques to find this little population. So I will be a little bit fast in the introduction. So macrophages, we can find macrophages from two different origins. We have the first origin that comes from the embryogenic phase, or from yolk sac, or from fetal liver, that you give rise to the tissue-resident macrophage in most of the cases. And the second wave came from the bone marrow. So progenitor monocytes can get in the circulation and then get in the tissues and be differentiated in different kinds of macrophages, like said by Diego now. And when you go to tumors, most of the works describing macrophage in tumors are associating macrophages to a protumoral role. And this revision is just to point out a different kind of roles that were associated to macrophage in tumors. And for example, the role of macrophage in suppressing T-cell responses and helping angiogenesis and promoting tumor metastasis and the modulation of matrix and so on. If you go to the patients using some classical markers like this one, CD68, CD14, 206, and CD163, you can see in red, you see the association of macrophage to a bad prognosis for men and women. And in green, we have just colorectal, but it's still very conflicting. And for other types of tissues, it's still ongoing. One big problem of these works is that the markers, most of the studies used, are shared by monocytes, macrophages, and dendritic cells. So meaning that sometimes we could probably overestimate the population of macrophages, and probably we are looking at a mixture of cells instead of looking for really macrophage-like cells. When I read in the lab, we had this kind of results that stuck me at the time. First, we have compared the frequency of CD14 positive cells in different tissues from breast cancer patients. And we noted that actually lymph nodes that are metastatic, so invaded by tumor cells, they present a higher percentage of CD14 positive cells in comparison to lymph nodes that are not invaded by tumor cells. Here we have an example for 4 lymph nodes from the same patients, and you can see here represented by EBCAN, so the expression of epithelial tumor cells, that more you have epithelial tumor cells, more you have also CD14 positive cells. So there's some kind of correlation between the accumulation of CD14 positive cells and the metastasis in these lymph nodes from breast cancer patients. And so at the time, we decided to answer one big question is to understand how tumor can drive or modulate the myeloid cell landscape, and mostly focus on the heterogeneity we can find in those tissues. To do that, we took advantage at the time, it was in 2017, for a technique that was emerging in the literature, is that single cell nanosec. So this is very representative, but the idea is we are able now to analyze each cell and to understand what is the transcriptome associated to each type of cells. Now, if you compare here in a tumor, for example, as Diego mentioned before, we have a mixture of many cells that come from epithelial tumor cells and stromal cells. And now with single cell, we can really dissect what kind of cell we have in those kind of tissues. So this somehow have changed a lot the way we see science and immunology. And we start to do experiments using tissues from patients. So what we did was we obtained blood, invaded lymph node, metastatic lymph nodes, or primary tumors. We processed those tissues, and we did a FACS sorting, most focused on disease and myeloid subsets, and we performed single-cell nanosec using the 10x genomic platform. Here are the results. Here's the representation. Each dot you see here is one cell, and we could define different clusters that make part of this mixture of tissues. Here we identify 6 different clusters. We have the classical CD14 monocytes. We have CD16 monocytes. We have a mixture of disease 1 and 2. We have some cycling cells and stressed cells. And we also have this population of macrophages that express mostly those genes. Here is the same representation by line plot. And the first conclusion we had at the time is that ApoE could be one marker that can be used to distinguish macrophage from the other subsets. So disease, monocytes, and other subsets do not express ApoE at the transcription level. So we decided to validate this using flow cytometry. We did a new mix of a new panel of antibodies trying to dissect the characteristics of those cells. So in this case, we use distinct types of tissues from breast cancer patients. We use CCR2 as a marker for monocytes and NAPOE as a marker for macrophages. And what we could see is the lymph nodes, when presenting a lower percentage of tumor cells, they still have more monocytes and macrophages. When you go for a lymph node with a high presence of tumor cells, we have an unbalanced part to have a more ApoE positive macrophages than monocytes, and this is reproduced by primary tumors. So somehow this accumulation of CD14 positive cells could be related to the accumulation of ApoE positive TAMs. So our next question was, we did a zoom in the subsets of macrophages, and we dissect these subsets, finding 3 other clusters inside the macrophage subsets. We are representing different colors. And we perform comparison analysis trying to dissect what kind of genes can define those macrophages. Here doing an analysis 2 by 2, so cluster 1 versus 2, 0 versus 2, or 0 versus 1. The first thing that came out is that cluster 2, so this little cluster here, express those genes, and folder 2 was one of the top genes expressed by this cluster, while cluster 1 and cluster 0 also express those genes represented here, with the TRIM2 as being one of the top genes expressed by both cluster 1 and cluster 0. And so we, at the time, defined 3 major subsets identified by single cell, one expressing high levels of folder 2, and 2 clusters expressing different levels of TRIM2 and high or intermediate level. So we decided to answer some key questions about the folder 2 macrophages. At the time, some papers describing TRIM2 were all published, and so we have a focus more related to folder 2 because at the time, nobody in the literature had really dissected the cells. So the questions we try to answer is first the spatial localization of those macrophages, so probably proposing that those macrophages could be a tissue resident macrophage. We also go for a mouse model trying to do a mechanistic work and also to answer the question if those macrophages could be conserved among between species. Then the function, so if those cells can crosstalk with other immune cells, and then the relevance of those cells for patients' outcome. So to dissect the tissue localization, this was performed in collaboration with Jérôme Galon. And so we performed a multiplex analysis using 2 distinct tissue microarrays from breast cancer patients. And what you can see here I circulated tumor cells that are in the middle. And you can see the tissue localization of both folder 2 macrophages or TRIM2 macrophages is really distinct when you look to many patients. First of all, in stroma, we didn't see really difference about infiltration of folder 2 or TRIM2 macrophages. While if we go to tumor nest, we can see that the great majority of cells that are in tumor nest are TRIM2 positive cells and not folder 2 positive macrophages. We also analyzed the distance of those macrophages to the tumor nest. So here is represented, and in red you have the curve showing the limit of distance of folder 2 macrophage to the tumor nest, and here with TRIM2 macrophages. And we measure the distance of those cells to the tumor cells. And what we look at is that TRIM2 macrophages are much more closer. Sometimes they form a barrier around surrounding the tumor nest, while folder 2 were much more spread in the stroma, like you see in the scene. So this was true for many breast cancer patients. We increased our analysis also looking for other kind of tissues, other kind of tumors, and most focused in folder 2 macrophages. We can see for primary breast cancer, for different subtypes, we still have much more folder 2 macrophages spread in the stroma. This was also true to distinct kind of tissues like glioblastoma, kidney, liver, melanoma, lung cancer. And also for some metastases we get the opportunity to have, we also have those folder 2 macrophages much more spread and in the stroma and far from the tumor nest in comparison with TRIM2 macrophages. Then we go for a mouse model. So we took advantage for this model of a spontaneous development, so MMTV and PYMT mice. We also did kind of the same protocol for tissue processing, and we sorted at this time lineage negative, positive lineage cells, and used a single cell transcriptome analysis. Here I go directed to the zone of macrophage population. And so we could find, again, our little population of folate2 macrophages, and also found two distinct populations of macrophage expressed in distinct levels of 6, 3, CR1. We performed different kind of analysis trying to compare the score of macrophages from mice and human. And we could see that folate2 macrophages from mice and from humans, they have the higher score of similarity comparing the distinct signatures those macrophages can express. One thing that's very important in our work is more a technical question, but was very key in our work, is that it was very, very difficult to have a TRIM2 expression using flow cytometry. We tried different kind of approaches, and actually we come back to our analysis and we found that CAD1 could be one marker that can replace TRIM2 both in mice and human, and folate2 is still validated in protein level also. So we went back to the patient samples. First, we wanted to quantify the folate2 positive macrophages. Here I'm showing you just a tumor, so a tumor that is not committed by tumor, that you have a great majority of macrophages that express folate2. And here, if you compare a tumor with more or less 2 centimeters, we can start to see that the loss of a folate2 in this haploid population. Here's the quantification, showing kind of the same thing. So metastatic lymph nodes with a high invasion by tumors lose the expression of folate2 among the total haploid positive cells, and tumors with different sizes also start to present a loss of folate2 proportion among haploid positive cells. We also try to validate this in mice, because in mice we can track tumor develop in different weeks. And here comparing a mammary gland that is not committed by tumor cells and a mammary gland committed by tumor cells, you can see the great majority of cells, macrophages gated on these molecules, are folate2 positive. When, if you look to mammary glands from PYMT mice in around week 12, you can start to see the appearance of an accumulation of these CAD1 positive cells. And here's the quantification showing kind of the same picture we saw in the patient. So this led us to know that folate2 macrophage is in the tissue before the tumor appearance in mice and in human. Then we go a little bit more in the function. So we perform a FACS sorting for NSEC and using different kind of markers. At the time, we sort monocytes, folate2 macrophages, and CAD1 or TRIM2 positive macrophages. What we saw, first of all, is that the transcriptome associated to folate2 macrophages is really different from CAD1 or from monocytes. While monocytes and CAD1 macrophages can share many of these features in terms of transcriptome, so suggesting probably those cells, CAD1 positive macrophages, can come from CCR2 monocytes. Then we look at genes that could be responsible for some role, and we found that folate2, genes expressed by folate2 could be related to wound healing, chemotaxis, and regulation of immune responses of cell activation. So it started to give us some idea of function of those folate2 macrophages. We performed some kind of correlation analysis using distinct data sets, public data sets. This I'm showing just with Metabrick. We are correlating distinct types of immune cells present in the tumors with folate2 gene, TRIM2 gene, or the signature, folate2 signature, or TRIM2 signature, based on all those markers we found in single cell. And what you can see, more red, you have more correlated are those populations. So folate2 gene or folate2 signature are positive correlated with NK, tissue-resident memory, CD8, TLS, cytotoxic cells, T cells, while TRIM2 do not correlate, or we don't have a score of correlation of TRIM2 with those cells. We validate using different kind of approaches. Here, I'm just showing immunohistochemistry and immunoconfocal analysis, showing the colocalization of folate macrophages with CD8 in distinct approaches. And we needed to have some kind of clues about the mechanistic or about the function of those folate macrophages. So we come back to the mice model, and we did a very classical experiment that was cited before by Pierre. And so we sorted folate macrophages or cadmium macrophages. We incubated those with ovapeptide, we wash it, and then we co-culture those with a specific OT1 positive CD8 cells. And what we saw was folate macrophages can induce a profile of activation of the CD8 positive cells by the loss of CD62L and expression of CD25. And also, those folate macrophages are able to induce a polyfunctional phenotype of the CD8 by the production of interferon gamma and granzyme B in this case. And here we are just quantifying the number of cytokines those cells can induce. And so we can see folate 2 can induce a huge proportion of CD8 that can express 4 or less cytokines at the same time. So showing that folate 2 are able to present antigens and to stimulate CD8 in vitro. But one thing that was really interesting for us is this kind of graphic here. When you isolate folate 2 macrophages from a healthy mammary gland, so without tumors, those macrophages are not anymore able to induce interferon gamma or granzyme B production. So it was intriguing at the time, so we decided to go a little bit further in this history. So what we did was we used the same mouse model, and we isolate folate 2 macrophages from healthy mammary gland, from small tumors or from medium tumors. And we perform a single RNA-seq bug. And what we observed, what is very nice, is that if you compare the transcriptome of folate 2 macrophages from a healthy mammary gland, small tumor, and medium tumor, we can see changes in the signatures or changes in the transcriptome of those folate 2 macrophages when the tumors start to progress. And so we want to understand what kind of signatures could be related to this kind of polarization phenotype. And when we compare healthy mammary gland, folate 2 from healthy mammary gland or for tumors, we could see that during a steady state, most of the genes were related to metabolic genes, while in tumors, folate 2 start to trigger a programming of immune cell stimulation due to different kinds of genes. So, and somehow the presence of tumors and the progression of tumors can be sensed by folate 2 macrophages, and macrophages start now to be like triggering the immune responses. And so what is the relevance of all this? We come back to the patients and we perform multiplex analysis now using the 2 cohorts and looking for folate 2. And in the 2 cohorts, we analyze in the dependent analysis the higher density of folate 2 macrophages were associated to a good prognosis. We were kind of obliged to validate this in 5 additional public cohorts. And not time to show it today, but you need to believe me. And also the same profile of CD8 density is kind of similar. So more you have folate 2, more you have CD8 in both cases. And it's the same that we found in silico. So to summarize, the working model we are now is monocytes characterized by those markers can get out from blood and enter in the tissues, acquiring this phenotype. I didn't show you, but we have an intermediate phenotype of macrophage expressing both monocytes and macrophages proteins at the same time. And we do believe those cells were giving rise to TRIM2 or CAD1 positive macrophages. And this is associated mostly to inflammation or to increasing of the tumor and progression of the tumor. And so macrophages expressing TRIM2 are mostly found in the tumor margin and the tumor nest, and probably their role is much more related to regulation of metabolism. This was described by other studies. In the other side, we have folate 2 macrophages that probably are coming from the embryonic phase, we really don't know the contribution of circulating monocytes to folate 2 macrophages. Those macrophages are much more found in the steady state and homeostasis. In tumors, they are much more in the stroma, not in the tumor nest. And what is interesting is that when you have tumor growing, we have a kind of reprogramming of these folate 2 macrophages to acquire an immunostimulatory phenotype. And we do believe that this could be associated to a better patient outcome. And I need to thank very much everybody. So our team in Curie, so Juliette was the PI of the study. Johan Misolo was our bioinformatics that did most of the analysis. Johan Gerbert was dedicated to the mice models. All this work was developed in the Eliane Piaggio's lab, and many members of the lab participate. Many people from Curie also participate, and many external collaborators, including Pierre and Jérôme, was possible to this work. And thank you very much for your time. I was kind of quickly, but probably.
Thank you, Rodrigo. Vanderson,
so
please.
For the survivors.
So this is very interesting. So once again, in which tumors do you think, instead of breast and colon cancer, 4R2 can have a role, Rodrigo? Which cancers did you already have some data?
Yeah, we have tested more in silico analysis using TCGA, so it's more a correlative analysis. So for lung, if I remember well, for lung, for kidney, the association is much more good prognosis. For many tumor types, we don't have a score of prognostic, so we have zero, neither positive, neither negative correlation, so it needs to be further investigated. And then also another thing we think is folate 2 macrophage is still a very heterogeneous subset. So I think we need to still go deep in the subsets of folate 2. For hematologic cancer, we never tested, if it was this your question. So we need to test for hematologic cancer the role of those macrophages. So for instance, we are sure in breast cancer, but the others we need to revalidate this.
And is there any relationship with FORWARD2 and Immunoscore and the infiltration of T cells?
Yeah. We can see a good correlation between folate 2 and CD8. So we performed this analysis by in silico, by immunoflow cytometry, and by immunohistochemistry, and there is a good association between folate 2 and CD8 in the tissues. So we found many clusters of folate 2 with CD8 in distinct type of tissues, and we do believe those cells can play a role in vivo. We do believe.
But which is the reason? Because when Jérôme showed, I was wondering which are the reasons that there are tumors that we have a lot of CD3, CD8, and the others not. Do you think it's something local, or there is one comment of Gilton showing that probably since you have a charge of bacteria inside, for example, the colon, this could be related to the immunogenicity of the cancer, but this is not the right thing if you think about breast cancer or other cancers not exposed to bacteria. Do you think is there any relationship with this?
It's a very interesting question, and I think we don't have the answer. For instance, I think it's much more related to the background of the individuals. So there are many works trying to correlate now age, stage, and health habits, but it's very complicated. You have some associations, like in this case, CD8 with folate 2, CD8 of disease. We have the TLS, the tertiary lymphoid structure that can appear in the tumor, near the tumor nest, and can be associated to a good prognosis. But I think the reason why we have patients with cold and hot tumors, for example, it's very complicated. I think I don't know if Professor Barbuto has other clues about this.
It's the dendritic cell. Dendritic cell.
Other questions? Barbuto questions. It's really a very nice work, Rodrigo. Congratulations.
Thank you.
Other comments?
Rodrigo, we have a question on YouTube from Enio. He asked you if you studied the biological role of FOLR2 for the macrophages. If you study the biological role of FOLR2 on macrophages.
The biological role. Yeah. So one thing that's very interesting is FOLR2 is a receptor of folate. There is, I think, 2 works trying to use FOLR2 as a receptor for delivery, and it's working very well. We can use this receptor to re-stimulate macrophages, but for instance, in our experience, we never tested what are the changings when you stimulate via FOLR2. So, for example, if they are changing the metabolism or if they're changing the profile of immune responses. In our case, we use FOLR2 much more as a marker, a surface marker, and not really, we didn't investigate really the function. So I know people in Paris, they continue to study this, trying to do knockout mice to address this question, but still FOLR2 is just a receptor. So we need to go further in the transcription of factors and try to deplete those cells very early in the development.
It would be interesting to look into hematological patients that have problems with folate.
Sure. I think Christophe wants to make a question.
Yes, Rodrigo, very nice work. I have one question regarding the link between these macrophages, the fact they are of good prognosis, and the immune infiltrate, and more specifically to tertiary lymphoid structure. Do you have, within TLS, I don't know if this has been looked in tumors, but in lymphoid structure in lymph nodes, you have tingible body macrophages, for example, which I believe are probably tissue-resident macrophages, and not coming from monocyte. Do you think do you have any link with TLS, to be simple?
Yeah, this is a very good question. We perform many stainings using immunofluorescence or immunohistochemistry, trying to see FOLR2 in those structures, and we could find FOLR2 sometimes surrounding, so giving a kind of support to the formation of some TLS, and also sometimes inside the TLS, we could find FOLR2 positive macrophages in low proportion. So at the time, we were a little bit, what we can do with this, and at the time, we removed from the history, but this is a field that we need to go further. So why, for some patients or for tissues, we have FOLR2 inside the TLS? So what's the role of those FOLR2 inside the TLS? And why sometimes the FOLR2 are near, so they are surrounding. One guess we have by looking at the transcriptome, those FOLR2, they produce and then they express a lot of chemokines, and distinct chemokines. And so
you mentioned that they are heterogeneous. So maybe there is different flavors of resident macrophages are very different from tissue to tissue. So if you have a lymphoid tissue within a tumor, maybe they adapt to this tissue.
Yeah, we have, when we look at breast and now in colorectal, we are looking a little bit, we can find FOLR2 near to vessels, associated to vessels. We can find FOLR2 spread on the stroma with some kind of homogeneous organization, and sometimes near the TLS. So one idea we have is probably we can try to look at the role of those subtypes of FOLR2 in the distinct localizations. For this, probably using some single cell spatial analysis or something like that. But yeah, we believe that we still have heterogeneity, and also among tumors. So if I go to a tumor that they are associated to bad prognosis, probably the role of those cells are completely different.
And I have just maybe you show it, but do you know if they express MHC class 2?
Yeah, they express and they present antigens via class 1 at least, but they express high levels of class 2. They are not the class 2 low macrophages in mycin and mycin.
Okay, and this is common property there. All FOLR2 macrophages are class 2 positive.
Yeah. One thing that is important to mention, probably, and also taking a little bit the work of Diego, is that we look at the classical M2 and M1 markers for these macrophages, and FOLR2 can express both markers. They express a little bit more 206 and CD163, but when you get the whole 206 population, just a minor population among 206 express FOLR2. So somehow we are going in another level of complexity by looking at another subset of what we expected, what we believed was the M2-like, but I think inside this M2-like, we have one subset that have this role, and we believe they are tissue resident.
Great. Any drug that increase the expression of FOLR2?
If there is a way to increase the expression?
Yes, any drug?
Yeah, drug I don't know, but we can treat those cells with MCSF, and we can see the higher expression of FOLR2. So there is an increase.
And with GM-CSF?
GM, I never saw. Just the works I saw was MCSF because people are looking for the M2-like. But we can try. We can try.
But you can change a cell that was FOLR negative to positive. Or you just increase the expression?
Because these works, I remember, came from monocytes. So monocytes, they don't express FOLR2 at steady state. And so when they are treated by MCSF, there is an increasing of FOLR2. But I never saw, for example, kind of a repolarization model where you take a high FOLR2 macrophages and try to repolarize to a low. But monocytes do not express FOLR2, so there is an increasing. And we don't know the cytokines. We don't know nothing about how the expression of FOLR2 is modulated, actually.
So you can induce it on monocytes?
Yeah, it's possible to induce.
Through CSF1?
Yeah. This, I mean, we never did, but there are 2 papers showing that. And we need probably to reproduce, but those 2 papers are kind of very, very well done. So,
okay.
Yeah.
And using folate.
Yeah. I mean, we are open a gate about questions we need to make now. I hope this work will be accepted. We are under revision now, but. I mean, let's see. Probably we hope to open a new gate of analysis by targeting FOLR2 macrophages. We can use folate, we can use cytokines or drugs to modulate FOLR2. But I don't think it's just the FOLR2 expression, but there is a whole transcriptome associated to this FOLR2 that we need to study.
Mm-hmm.
Do we have time for one more question from YouTube?
Yeah, please.
Okay, Elias.
The survivals.
Yeah, there's some still. So Elias Almeida is asking you if you have performed any assay in terms of mitochondrial metabolism. He wonders if these cells could suffer metabolic reprogramming due to glutamine from carbon metabolism.
That's a great question. We really don't know. We all have, when we analyze the FOLR2 from healthy tissue, so no tumors from the mammary gland for mice, they express a lot of genes related to metabolism. So the only clue we have is that probably in steady state, they play a role in metabolism and they can suffer some kind of stimulation from the tissue. But also we have the tumors appearing, we have a completely skewing in this transcriptome. And so this could be a good question to analyze what the metabolism compromised in this FOLA2. The one thing is very complicated in human is that the percentage, the frequency of those FOLA2 are very tiny. So it's very hard to sort those cells from tissues, and it's why we needed to go to mice to have more cells to work. And so this is a limitation. So we can probably try to find a model or tissue where you have much more FOLA2 to work with.
Spleen.
Yeah, probably. Great. We need to explore, yeah.
Wonderful.
So thank you very much. We need to close. I think I really enjoyed. I think you too. Great presentations, our invited speakers, and we need to collaborate now. We need to establish some collaborations about DCs and T cells and macrophages, and to form our.
Thanks to Christophe that remained till the end. That's
yeah, it's true. Thanks for staying here.
I think Pierre is here as well.
Yeah, Pierre is here.
Yeah. Oh, I don't see him.
Pierre and Christophe. So I would like to thank Rodrigo and Barbuto for this very nice workshop. This is the 5th workshop, and I really appreciated the high quality of the speakers, and it was a great day. I hope that the 6th workshop, we can already organize. I think it will be in March, so everybody's already invited to participate. We have just to choose, the subject of the next workshop. But and we would like to have, of course, suggestions. If the people here would like to, to focus on some kind of subject related to our laboratory, I think it's very good. Thank you very much, everybody. Barbuto and Rodrigo just finished.
No, I just have to thank and let Rodrigo finish. He was the main organizer. Actually, he's the guy.
Okay, so thank you very much. It was a pleasure for us, and we enjoyed this meeting. I hope to see you soon. Thank you very much, and good weekend for everybody.
Thank you.
Thank you. Bye-bye.
Bye.
Bye.
Ciao, ciao.
Bye.
Bye-bye, Pierre. Bye, Christophe. Bye, Vanderson.